Did you hear these concepts, declarative memory and procedural memory? Suppose like if I ask you, who is the first president of India, or who is the first woman prime minister of India, what do? You try to remember, you try to see where you have studied, in which class, where you have read, which newspaper or which general knowledge book, and you try to give the information. So this information is about the world, these are the facts, the general knowledge which you possess, the language, the grammar, irrespective of the language, all these are the facts. This is called declarative memory. On the other hand, the procedural memory is called as non-declarative also. This procedural memory contains your own skills, like your riding of a bicycle, or if you know the swimming. So all the skills that you have, the abilities that you have in performing a particular behavior, particular art, all these things come under the procedural skills. Now to understand about this long-term memory modules, look at this slide. The slide shows different types of long-term memories. As I have already mentioned, it can be divided into declarative, which is explicit, that is, which can be seen by the others and which you are conscious of. And the other is procedural, call it as non-declarative, and it is implicit. It is not done consciously, that is, you may not be able to remember this information consciously, but when the time demands, you try to use that information. The declarative memory can further be divided into semantic memory and episodic memory. So semantic memory is nothing but the facts about your general knowledge. Whatever examples I gave you just a few minutes back about your tables, about your mathematical sums, or about your general knowledge, all these things come under the semantic memory. Then the declarative memory can also be divided into episodic memory. Episodic memory indicates the significant experiences that happen in your life. When did you first join in the school? What was your experience? Or the day you have got your gold medal, the day you have graduated, all these things come under the episodic memory. Similarly, the procedural memory can be called as skills, that is motor and cognitive, all the skills that you have learned in playing the games or in riding the vehicles, and then your personal characteristics. So we can say the declarative memory contains the information about the things, while procedural memory contains the information about how to do the things. And this basic distinction between procedural and declarative memory is given by a researcher called Tulving. Another model of memory which is in the contemporary approaches is associative models of memory. So this model was postulated by Collins and Quillen and Collins and Loftus. This model suggests that memory consists of mental representations of clusters of interconnected information. According to this model, if you think of one particular concept, you are able to generate more of the information which is interrelated to that concept. For example, the moment I ask you the meaning of an animal, what comes to your mind? Animal has 4 legs, or animal can be carnivorous, herbivorous, or animals can be wild, domestic. So all this information is triggered by just one word given by me, that is animal. Similarly, if I say bird, you can think of a robin is a bird, it eats worms, so any information related to that. So activating one memory triggers a large amount of information related to the similar concepts, and this is known as spreading activation model. Associative memory also accounts for a priming effects. It is a phenomena in which exposure to a word or a concept later makes it easier to recall related information. This priming effects occur when a person has no conscious memory of the original word or the concept. Then there is another approach called levels of processing theory. According to this theory, depending upon at what level the information is encoded, the memory would be effective. So this model is postulated by Craik and Lockhart. According to this model, there are 3 levels. One is shallow level, where the shallow processing takes place. One is intermediate level, where intermediate processing takes place, and the third level is deep level, where deep processing takes place. So, what type of encoding occurs at these levels? At the shallow level, only the physical structure of the stimulus is encoded. For example, if I show you an advertisement paper and say the term Reliance is typed in capital letters or in small letters. So, the moment you see the advertisement and then immediately respond to me whether the word is in capital or small letters correctly, that is, you have encoded that information at the shallow level. At the intermediate level, the information will be encoded in a phonemic form, that is, whether any rhyming takes place. And at the deeper level of processing, the information is encoded in the form of semantic form, that is, you are trying to understand the meaning of it. Suppose if I say truck, and then if I ask, does the word truck belong to the furniture category? So, what would be your answer? If it is not a furniture category, you would say no. If it is a furniture category, you would say yes. So, to understand the word furniture and the truck, you should have some kind of a semantic information there. Otherwise, you cannot give the accurate answer. So, according to this model, the deeper the level of processing, that is, if the information is encoded in a semantic form, only then the information can be retrieved in a more effective way. So, once we have understood about encoding and storage, what is about retrieval? Retrieval is getting the information out of our storehouses. So, here retrieval can be understood through different methods like recall, recognition and relearning. So, how will a person's memory is tested, that is, whether the person could retrieve the information in an effective way or not? One simple way is your exam. Whatever you have studied throughout the academic year, your final exam or any other exam for that matter proves whether you could recall the information that you have studied throughout the year or not in an effective manner or not. So, I am asking a question and you are trying to answer, that is recall. Then recognition. When you answer your multiple choice questions where you are given 4 options and you have to choose the correct answer for your question. So, this is recognition. There are certain clues which are available. If you have studied, if you know the correct answer, it would be easy for you to pick out the right answer from the list. Then the other one is relearning. Relearning is first time you have studied one lesson or say a poem, you have taken 5 trials to read it. After a week or after 10 days, when you are studying the same poem again, how many trials you have taken? Same trials, same number of trials, or you have reduced the trials. So, this can be calculated by using a simple formula like original trials minus relearning trials by original trials into 100. So, this gives you the how much of the percentage of savings occurred in terms of your trials. So, these are the 3 methods through which we can try to assess the retrieval process of the human being. There are other kinds of memory like flashbulb memory, where we try to remember in an effective way some memories which are very important, which are very specific and which are very surprising events. They are so vivid that they are like a snapshot of the event. For example, it could be you have witnessed an accident long before, but still you are able to say that it is very fresh in your mind. That is something like a flashbulb memory. Or the first day you have met your roommate, that experience is still fresh in your memory and you are able to say when and where you could meet the person. However, all the time the details may not be accurate, though they are easily retrieved. Then another important memory is autobiographical memory. It is something like information about our own episodic events of the life. However, sometimes it may contain the information which you do not want to remember because it is incompatible with your present. That is, you are adjusting to the life in an effective way, but you do not want to remember some of the incidents that have happened during the childhood as they were very insulting experiences. Sometimes we tend to forget what is incompatible to deal in the present. So, this is all about our human memory, about the different processes, about the different storages and different kinds of memory.
खूबसूरती और चमत्कारों से लदी पड़ी हैं प्रकृति की सारी सौगातें, और उद्देश्य होता है हमेशा सबके अच्छे के लिए। पर प्रकृति के एक चमत्कार हम मनुष्यों ने हमेशा दूसरे चमत्कारों को अपनी उपयोगिता के हिसाब से श्रेणीबद्ध किया है। वनस्पति भी इससे अलग नहीं। दरअसल सभी वनस्पतियों में कुछ गुण तो कुछ उपयोग छिपे रहते हैं। अधिकांश में औषधीय गुण बहुत सारे हैं, तो कुछ वनस्पतियों में पाए जाने वाली मादकता को हमने पहले स्थान पर रखा और इसकी औषधीय प्रकृति और अन्य उपयोगों को दूसरे नंबर पर। ऐसी ही एक वनस्पति है तंबाकू।
तंबाकू की कहानी और इसकी विकास यात्रा काफ़ी पुरानी और रोचक है। अमेरिका की खोज के बाद जब यूरोपियन लोग अमेरिका गए तो उन्होंने वहाँ के मूल निवासी रेड इंडियंस को तंबाकू का सेवन करते हुए पाया। ये लोग तंबाकू के धुएं को सांस के द्वारा अंदर खींचने के लिए एक विशेष प्रकार की नली का उपयोग किया करते थे। इसे ये लोग टबैको कहा करते थे। बाद में तंबाकू को ही टबैको कहा जाने लगा। अमेरिका से लाए गए तंबाकू के पौधे पश्चिम यूरोप के देशों में उगाए गए और फिर 17वीं शताब्दी में पूरे यूरोप में तंबाकू खाने का रिवाज शुरू हो गया। यूरोप के बाद एशियाई देशों में और अन्यत्र देशों में भी इसका प्रचार शुरू हो गया। भारत में तंबाकू का पौधा पुर्तगालियों के द्वारा अकबर के शासनकाल में लाया गया और बस यहीं से बहुत ही तेजी से यह पौधा पूरे भारतवर्ष में मशहूर होता चला गया।
तंबाकू के पौधे की जाति का नाम निकोटियाना है, जो कि पुर्तगाली जॉन नाइकट के नाम के कारण रखा गया। उन्होंने इस पौधे को पहली बार फ्रांस में परिचित कराया। वानस्पतिक रूप से निकोटियाना जीनस के इस पौधे को सोलेनेसी फैमिली के अंतर्गत स्थान दिया गया है। इस पौधे की लगभग 50 प्रजातियां ज्ञात हैं, जिनमें से 2 प्रजातियां निकोटियाना टेविकम और निकोटियाना रस्टिका व्यावसायिक रूप से महत्वपूर्ण हैं। तंबाकू का ये पौधा शाकीय और 1 वर्षीय पौधा है, यानी ये अपना जीवन चक्र 1 वर्ष में पूरा कर लेता है। इस पौधे की जड़ें लंबी और रेशेदार होती हैं। तना सीधा खड़ा और गोल होता है। तने की और पत्तियों की सतह पर छोटे छोटे रोम पाए जाते हैं, जिनसे एक चिपचिपा पदार्थ रिश्ता रहता है। पौधे की ऊंचाई 3 से लेकर 6 फीट के लगभग होती है। पत्तियां लंबी और तने के ऊपर एकांतरित क्रम में लगी रहती हैं। पत्तियां अंडाकार और पर्णव्रंत रहित, यानी बिना किसी व्रंत के सीधे तने से लगी रहती हैं। पुष्प गुच्छों में लगे रहते हैं, जिसे पुष्पक्रम कहते हैं। पुष्प में पुष्प व्रंत पाया जाता है, यानी पुष्प व्रंत के माध्यम से शाखा पर लगा रहता है। सबसे बाहर की तरफ बाह्य दल होते हैं, जिनकी संख्या 5 होती है, जो कि आपस में एक दूसरे से जुड़े हुए होते हैं। इसके बाद दल होते हैं, जिनकी संख्या भी 5 होती है। ये दल आपस में मिलकर ट्यूब जैसी संरचना बनाते हैं। पुष्प में अंदर की तरफ 5 पुंकेसर यानी नर जननांग पाए जाते हैं, जो कि आधार पर दल के साथ जुड़े होते हैं, मतलब दल लग्न होते हैं। पुष्प के मध्य जायांग स्थित होता है, जो कि पुष्प का मादा प्रजनन अंग है। इनके अंडाशय में कक्षों की संख्या 2 होती है और जरायुज पर बहुत सारे बीजांड लगे हुए होते हैं। ये अंडाशय बाद में फल में परिवर्तित हो जाते हैं। पुष्प का बाकी भाग सूख कर झड़ जाता है। तंबाकू के रासायनिक संगठन में बहुत सारे एल्केलॉइड्स पाए जाते हैं, जिनमें निकोटीन, निकोटेनिन, निकोटेन और निकोटेलीन प्रमुख हैं। दरअसल जब तंबाकू की पत्तियों का धूम्रपान किया जाता है, तब ये निकोटीन विघटित होकर पायरिडीन, फरफ्यूरल, कोलिडीन, हाइड्रोसायनिक अम्ल और कार्बन मोनोऑक्साइड में परिवर्तित हो जाता है। ये पदार्थ जहरीले और नुकसानदायक होते हैं।
जब हम निकोटीन का सेवन करते हैं नशे के रूप में, तो हमारे जो स्मूथ मसल्स और के ऊपर जो एक्शन होता है, उससे जो एक स्टिमुलेंट एक्शन मिलता है, उसके लिए इसका उपयोग किया जाता है। बट जब लंबे समय तक हम इसका उपयोग करते रहते हैं, तो उसके कई दुष्परिणाम हमारे सामने आते हैं और स्मोकिंग की आदत हो जाती है, उसके ऊपर डिपेंडेंस होता है और उसके सारे दुष्परिणाम जो हैं वो हमें प्राप्त होते हैं। स्मोकिंग की जब आदत डेवलप हो जाती है, तो उससे कई तरह की समस्याएं होती हैं। एसिडिटी है, हाइपरटेंशन है, क्योंकि इससे गैस्ट्रिक सिक्रीशन वगैरह भी बढ़ता है। उससे निजात पाने के लिए एक डी एडिक्शन प्रोग्राम चलाना जरूरी है। इसके लिए निकोटीन के ट्रांसडर्मल पैचेस बना के और पेशेंट को दे दिए जाते हैं, या च्युइंग गम में भी निकोटीन इनकॉर्पोरेट करके पेशेंट को दी जा सकती है। इससे निकोटीन स्लो डोज में पेशेंट को लगातार मिलती रहती है और उसकी जो तंबाकू की ज़रूरत है वो धीरे धीरे कम होती जाती है। और इस तरह से नशा करने से जो एक अभियान होता है, नशा मुक्ति अभियान, उसमें निकोटीन का उपयोग किया जा सकता है।
यह निर्विवाद सत्य है कि आयुर्वेद में तंबाकू का उपयोग नशा के बतौर न होकर औषधि के उपयोग से जब होता है तो वो गुणात्मक है। आयुर्वेद में कहा भी गया है कि पंचभूतात्मकम विश्वम योजकस्तत्र दुर्लभा, जो विश्व में 5 तत्वों से बनी जो भी वस्तु है वो प्रत्येक वस्तु औषधि है, लेकिन हम उसका कैसा उपयोग करते हैं इस पर वो उसका उपयोग बनता है। जैसे हमने तंबाकू को ही लीजिए, तंबाकू को सूजन पर लगाने से सूजन घटती है। एक शोधहर योग तंबाकू में है, तंबाकू के पत्र पर शिलारस लगाकर थोड़ा उष्ण करके उसको यदि वृषण पर बांध लिया जाए तो वृषण में पानी भरता है, वो सूख जाता है बिना सर्जरी के, बिना ऑपरेशन के वो पूर्ण रूप से स्वस्थ हो जाता है। वैसे ही 50 ग्राम खड़िया मिट्टी और 50 ग्राम तंबाकू को पीस कर और उसे थोड़ा सा गर्म करके उसको वृषण पर लगा दिया जाए तब भी पानी सूख जाता है। इतनी गुणकारी अनुपम औषधि तंबाकू है।
भारतीय अर्थव्यवस्था में तंबाकू और इसकी खेती का एक महत्वपूर्ण स्थान है। तंबाकू की फसल सामान्यतः शीतोष्ण कटिबंधीय फसल है और इसे हमारे देश में रबी फसल के रूप में उगाया जाता है। इससे हमारे देश को लगभग 3000 करोड़ रुपए का राजस्व प्राप्त होता है। इस तरह से हमारा देश विश्व में अमेरिका और ब्राजील के बाद तीसरा सबसे बड़ा उत्पादक देश है और 8वां सबसे बड़ा निर्यातक भी। इसका अर्थ है कि हमारे देश में इसका सेवन बहुत अधिक है। इसलिए तो इसकी खेती लगभग 70,000 किसानों और 50,000 व्यापार से जुड़े लोगों को रोजगार मुहैया कराती है। परंतु कहीं रोजगार के चक्कर में या आर्थिक लाभ के चक्कर में हमारे देश का भविष्य संकट में तो नहीं? कहीं हमारे देश की युवा शक्ति तंबाकू के धुएं में खो तो नहीं गई? दरअसल तंबाकू में पाए जाने वाला अल्कलॉइड हमारे डाइजेस्टिव और सर्कुलेटरी सिस्टम को बुरी तरह गड़बड़ा देता है। इसके सेवन से हमें मिलता है कोरोनरी ब्लॉकिंग, हाइपरटेंशन, ब्रोंकाइल इन्फेक्शन आदि खतरनाक और भयानक रोग। कीमत और भी चुकानी पड़ सकती है यदि आप शिकार हो जाएं जानलेवा मुंह के या फेफड़ों के कैंसर से। हम ये क्यों नहीं समझ पाते कि यदि प्रकृति ने हमें तंबाकू जैसी वनस्पति दी है तो हमें विवेक भी दिया है। हम क्यों थोड़े से नशे के लिए अपनी जान दांव पे लगाना चाहते हैं और बदनाम करते हैं प्रकृति की देन को? प्रकृति की तो सारी सौगातें बहुत ही अच्छी हैं और इन्हें अच्छा ही रहने दीजिए।
students, welcome to the e-content lecture series on environmental sciences. Today, we will discuss global warming. Global warming refers to the increase in the average temperature of the atmosphere, oceans, and land masses. It is one of the greatest crises faced by mankind. Well, this problem is not only global in nature but potentially threatens the survival of life on Earth. The Earth's atmosphere acts as a greenhouse, wherein the shorter wavelength solar radiations of higher energy reach onto the Earth. Some of these radiations are reflected back by the atmosphere, but about half reach the Earth's surface, thereby heating it. And in response, the longer wavelength infrared radiations, also called as heat radiations, are radiated back. Most of these radiations, instead of being radiated back 100%, are retained by the atmospheric greenhouse gases. The greenhouse gases include carbon dioxide, methane, nitrous oxide, hydrochlorofluorocarbon, and chlorofluorocarbons. The greenhouse gases absorb infrared radiations emitted by the Earth's surface, atmosphere, and clouds. These gases in turn emit radiations in all directions. Thus, they trap heat within the atmosphere. This phenomena is termed as greenhouse effect. The greenhouse effect is responsible for maintaining the Earth's temperature at an average of 14 degrees Celsius. Without this effect, the Earth's temperature would have been minus 18 degrees Celsius. Today, the major area of concern is the increasing anthropogenic interferences that has disturbed energy budget of the Earth. The problem is not only of the gradual warming of atmosphere via enhanced greenhouse effect, but also due to the fact that greenhouse gases have long residence time in atmosphere, which envisages that despite the possibility that their emissions can be controlled, they could remain suspended in the atmosphere over a prolonged period of time. Now, the plausible causes of global warming. First of all, natural causes. According to skeptics, global warming is not human-induced. To them, it's natural on geological time scale. Many reasons have been put forth to support this view. First, the Milankovitch theory, which suggests that it's the normal cyclic variation in the Earth's orbital characteristic that is probably responsible for present climate change. The insolation from the Sun varies with three cyclic events known as eccentricity, secondly, Earth's rotation on its polar axis, and lastly, obliquity. Then, the Earth's natural cycle theory. Over the last few million years, the Earth's surface temperature has undergone natural fluctuations, expressed as ice ages. During a glacial period, the soil of Earth are fertilized by the ground-up mineral matter, so that at the beginning of interglacial period, the soil is fertile enough to support all plant life. That results in low carbon dioxide level. But towards the end of interglacial period, all trees and plants die off. As the mineral nutrients in soil are exhausted, the vegetation recedes. Less carbon dioxide is removed from the atmosphere, therefore its concentration in atmosphere increases. Immediate result of this increased concentration is the enhanced greenhouse effect. According to them, keeping in view the historic trend, the present global warming will trigger a new ice age. Lastly, changes in solar activity. The output of energy from sun is much more variable than expected. The sun's energy received at the top of Earth's atmosphere has been measured by satellites since 1978. Measurements made during 1980s showed a decrease of 0.1% in the total insolation received by the Earth over just 18 months' time. During this period, a maximum sunspot activity was observed, which led to decrease in solar activity. Now, global warming as a result of anthropogenic pressures. The alarmists are of the view that global warming is as a result of following factors. Firstly, changes in land use patterns. Different types of surfaces have different albedos or reflectivities. Deforestation and unsustainable urbanization has increased the albedo of Earth by approximately 0.006 over the last 1,000 years, leading to a change in radiative forcing by about 0.01 watts per meter square per decade. Then, increased emission of greenhouse gases. The increase in carbon dioxide level is almost 40% since pre-industrial times. The natural sources of methane have been intensified further by human activities. The concentration of methane is now more than 2.5 times the pre-industrial levels. Concentration of nitrous oxide has risen approximately 18% since the start of the Industrial Revolution. Also, tropospheric ozone, which has a short atmospheric lifetime, is a potent greenhouse gas. The chlorofluorocarbons not only destroy the stratospheric ozone but also contribute 15 to 25% to global warming. Also, chlorofluorocarbons, that is CFCs, hydrochlorofluorocarbons, that is HCFCs, hydrofluorocarbons, perfluorocarbons, and sulfur hexafluoride are together called F-gases. These are often used in coolants, foaming agents, fire extinguishers, solvents, pesticides, and aerosol propellants. These F-gases have a long atmospheric lifetime, and some of these emissions will affect the climate for many decades or centuries. Effects of global warming. Today, global warming is fast catching our imagination, and its effects are on every aspect of life. However, these effects are more severe than ever thought before. The first and foremost effect is the melting of glaciers. It will lead to rise in the sea level, which will lead to flooding, and this will in turn create havoc, as it will endanger several species of plants and animals, thus hampering the balance of the ecosystem. Moreover, these large glaciers reflect light back into the space, and with their meltdown, Earth will further be warmed. Rise in sea level. Coastal areas and islands are among the most densely inhabited areas in the world. Their proximity with sea makes them more prone to adverse effects of rising sea level, such as loss of land due to inundation, flooding, erosion, saltwater intrusion, and loss of fisheries and aquaculture. The IPCC projects that global warming causes thermal expansion of water, which will be the main component of expected sea level rise in the 21st century. Loss of biodiversity. Extinction of species due to global warming may either be due to loss of habitat, inability of organisms to adapt to rapid climate change, and lastly, climate change harbors pathogens against which organisms have no immunity. For instance, global warming has boosted fungal epidemic in midwife toads, Alites obstetricans of Spain, and harlequin frog species Atolopus of Costa Rica. The chytrid fungus Batrachochytrium dendrobatidis is a fatal pathogen of amphibians that interferes with their ability to control water loss. Coral bleaching. Warmer sea surface temperatures lead to coral bleaching. which is whitening of coral caused when it expels a single-celled symbiotic algae called zooxanthellae. These are expelled when the coral is under stress from environmental factors such as abnormally high water temperatures or pollution. Since the zooxanthellae help coral in nutrient production, their loss can affect coral growth and make them more vulnerable to diseases. Major bleaching events took place on Giant Barrier Reef in 1998 and 2002, causing a significant die-off of corals in some locations. Ocean acidification. The oceans serve as a sink of carbon dioxide, but increased level of it has led to ocean acidification. As carbon dioxide dissolves in seawater, carbonic acid is formed, which has the effect of acidifying the ocean, measured as a change in pH. The uptake of carbon emissions since the year 1750 has led to an average decrease in pH by 0.1 units. Projections in the special report on emission scenario by IPCC suggests a further reduction in the average global surface ocean pH of between 0.14 and 0.35 units over the 21st century. Diseases. Distribution of pathogens was earlier restricted due to seasonal temperature variations, but rise in the temperature and increased moisture has resulted in their extended distribution, leading to higher rates of infection. The major reasons for such outbreak is the warmer temperature provides conducive habitats for disease-causing mosquitoes, thus causing increase in transmission of infections via these carriers. World Health Organization estimated that the total attributable mortality was about 0.2% of deaths in 2004. Of these, 85% were child death. Increase in extreme events. A connection has been found between global warming and changes in the intensity, frequency, and the extent of disturbances such as fire, cyclones, drought, and floods. For instance, the hurricane frequency in Western Atlantic has increased to 3% since 1970. The hurricanes belonging to Category 4 and 5 on Saffir-Simpson scale are frequently occurring from the last 30 years, according to IPCC 4th Assessment Report. Also, El Niños are occurring after a period of 2 years due to global warming instead of a normal gap of 4 to 5 years. Despite the fact that global warming has adverse impacts, some researchers argue that increase in carbon dioxide level will have benefits that would outweigh any of its harmful effects. As we know, carbon dioxide is an essential raw material for all primary producers. The rise in carbon dioxide availability directly impacts photosynthetic processes in plants, evoking a wide range of physiological and morphological responses. Some scientists are of the opinion that doubling the concentration of carbon dioxide would increase plant productivity by 1/3. Due to these benefits, commercial nurseries use carbon dioxide enhanced environments to grow plants. However, the fact remains that plants cannot live on carbon dioxide alone. They get their bulk from more solid substances like water and organic matter. Also, soil conditions will not necessarily favor their growth even at optimum temperatures. Too high concentration of carbon dioxide causes a reduction in the nutritional quality of some crops. Researchers have found that the rising temperatures may boost the foraging by insects. The worst problem by far is that increasing carbon dioxide will increase temperature throughout the earth. This will make deserts and other types of dry lands. While deserts increase in size, other ecozones, whether tropical, forest, or grassland, will migrate towards the poles. Strategies to combat global warming. All the nations face the risk of global warming. although some might face catastrophic losses before others. In order to counteract the potential of massive risk, 3 basic strategies have been put forth. First, the waiting strategy, which envisages that before taking any substantive measures to combat warming, we should wait, since all the data is not yet in. However, such an approach of taking no action at all would be an unacceptable risk, as it might be found that the problem is real. The steps that would be taken for its mitigation then would be costly and painful than the measures that could be taken at the earlier stages. The worst case scenario strategy says that we should assume that the most extreme predictions are valid and act accordingly. This encompasses taking every possible step like decreasing fossil fuel burning, halting deforestation, encouraging reforestation, etc. The compromise strategy says that we should follow a path that is environmentally safe and promotes general well-being. It advocates a middle-of-the-road approach. The focus of it is to develop policies, increase conservation, raise energy efficiency, hamper use of fossil fuels, and lay more stress on sustainable use of renewable resources. One way to discourage use of fossil fuel is to impose carbon tax on their consumption. A carbon tax is levied in proportion to the amount of carbon emitted during consumption of the fuel. Such a tax will encourage development of non-carbon emitting technology. Another suggestion is to institute a standardized system of emission permits, both at a national and global level, that would allow organizations to emit certain amount of greenhouse gases into the atmosphere. Lastly, controlling deforestation will not only abate greenhouse effect via providing sinks that absorb carbon dioxide, but will also preserve biodiversity, ameliorate climate extremes, and help to protect soil. Hence, it can be concluded that even though there is less scientific and political consensus concerning the severity of the threats of global warming, still, in case of greenhouse gas emission, every contribution towards the ultimate solution counts, as the beauty of all actions taken will offer numerous other benefits. This is a classical example of win-win strategy. That is all we have for you in today's session. Thank you for watching.
प्रकृति प्रदत्त संसाधनों में जल एक ऐसा संसाधन है जो सभी जीव जंतुओं के अस्तित्व को बनाए रखने के लिए नितांत जरूरी है। यदि ये कहें कि जल ही जीवन है तो कोई गलत बात नहीं होगी। जल प्राप्ति के स्रोतों में वर्षा का प्रमुख स्थान है। वर्षा पूर्णतः एक वायुमंडलीय घटना है। वायुमंडल के विभिन्न घटकों जैसे ताप, नमी, वायु दाब, वायु गति और वायु दिशा के आधार पर ही वर्षा का सृजन होता है। ग्रीष्म काल में सूर्य की प्रचंड गर्मी पाकर महासागरों का जल बड़ी मात्रा में वाष्प बनकर आसमान में पहुंच जाता है, जो बाद में घनीभूत होकर नमी और ठंड वाले स्थानों में वर्षा में परिवर्तित हो जाता है।
जून के महीने में जब सूर्य मध्य भारत स्थित कर्क रेखा पर लंबवत होकर चमकता है तो उत्तर भारत भी तपने लगता है। ऐसे में होता यह है कि उत्तर पश्चिमी भारत में निम्न वायुदाब का केंद्र निर्मित हो जाता है, जबकि इसी दौरान हिंद महासागर में उच्च वायुदाब का केंद्र बन रहा होता है। इससे होता यह है कि उच्च दाब वाले क्षेत्र यानी हिंद महासागर से अत्यधिक गर्मी के कारण उठने वाली वाष्प और नम हुई हवाएं निम्न वायुदाब वाले क्षेत्र उत्तर पश्चिमी भारत की ओर वायु शून्य हुए इलाके की तरफ तेजी से दौड़ने लगती है और तब शुरू होती है भारत में बारिश।
निरंतर बढ़ते औद्योगिकीकरण और शहरीकरण के कारण पर्यावरण प्रदूषित हो रहा है। इसका असर वर्षा पर भी पड़ा है। वास्तविकता यह है कि आम वर्षा भी अब शुद्ध नहीं रही। वैसे तो इसमें कार्बन डाइऑक्साइड की थोड़ी सी मात्रा घुली हुई होती है, जिससे उसका पानी अम्लीय होता है, लेकिन इस पानी से कोई नुकसान नहीं होता है, क्योंकि पानी में कार्बन डाइऑक्साइड घुलकर कार्बोलिक अम्ल बनाती है, जो कि एक कमजोर अम्ल है। असामान्य स्थिति तो तब उत्पन्न होती है जब बड़े बड़े उद्योगों और वाहनों की सल्फर डाइऑक्साइड, नाइट्रोजन ऑक्साइड जैसी गैसें वायुमंडल में जाती हैं। अनुकूल परिस्थिति में जल वाष्प और ऑक्सीजन से अभिक्रिया करके ये गैसें सल्फ्यूरिक तथा नाइट्रिक अम्ल बनाती हैं। ये अम्ल बाद में वर्षा के साथ मिलकर पृथ्वी पर आते हैं। इस वर्षा को एसिड रेन या अम्ल वर्षा कहते हैं।
कल कारखानों की चिमनियों से जो गैसें बाहर निकलती हैं, उनमें सल्फर और नाइट्रोजन के ऑक्साइड भी सम्मिलित होते हैं, और ये ऑक्साइड जब वातावरण में फैलते हैं तो पानी में क्रिया करने पर अम्ल बनाते हैं। ये अम्ल होते हैं सल्फ्यूरिक एसिड और नाइट्रिक एसिड। ये एसिड बहुत स्ट्रांग एसिड होते हैं। कहने का मतलब ये कि इनमें हाइड्रोजन आयन प्रदान करने की क्षमता अद्भुत होती है, और ये अम्ल जब सामान्य वर्षा के साथ नीचे आते हैं तो जिस किसी भी पदार्थ या जीवित वस्तु के संपर्क में आते हैं तो उनके साथ रासायनिक क्रिया कर अपने दुष्परिणाम छोड़ते हैं।
साधारणतः आम वर्षा का पीएच लगभग 6 होता है, यानी कि थोड़ा अम्लीय। लेकिन जब कार्बनिक संयोजन धातु गलन यंत्रों, आणविक संयंत्रों, थर्मल पावर संयंत्रों द्वारा सल्फर डाइऑक्साइड और नाइट्रोजन ऑक्साइड की भारी मात्रा वायुमंडल में प्रवाहित होती है, तो वर्षा के जल का पीएच 4 से भी कम हो जाता है। जितनी अधिक मात्रा में सल्फर डाइऑक्साइड और नाइट्रोजन ऑक्साइड वायुमंडल में पहुंचेगी, वर्षा में जल की अम्लीयता उतनी ही बढ़ेगी। अम्ल वर्षा पर्यावरण के संतुलन पर असर डालने वाली एक घटना है। इसका असर जीवों और अजीवित पदार्थों दोनों पर पड़ता है।
मुझे केरल में आलवाई के पास उद्योग मंडलम रासायनिक संयंत्रों के संकुल में तेजाबी वर्षा का एक अनुभव अभी भी याद है। यह शायद अगस्त-सितंबर 1969 अर्थात आज से लगभग 30 साल पहले की बात है। मैं वहाँ की एक रासायनिक फैक्ट्री में कार्यरत था। हल्की हल्की बारिश हो रही थी, फुहार थी बारिश की, वर्षा नहीं हो रही थी, पानी बह नहीं रहा था, लेकिन हल्की हल्की फुहार थी। अचानक हमारी आंखों में जलन होने लगी। बारिश की बूंदों के साथ कुछ बूंदें जो हमारे कपड़े पर गिरी तो कपड़ों पर भी झुलसन जैसे कुछ चिन्ह बन गए। हमें ऐसा लगा जैसे कपड़े में छेद हो रहे हों। यह स्थिति कुछ समय तक रही। वातावरण में दम घुटने जैसी भी हमने महसूस किया। थोड़ी देर में बारिश तेज होने लगी, तब वो सब कुछ साफ हो गया। दम घुटने वाली जो बात थी वो भी नहीं रही, और तेजाबी वर्षा का जो असर था वो भी बाद में हमें नहीं महसूस हुआ। दरअसल उद्योग मंडल समुद्र किनारे बसा होने के कारण वहाँ क्लोरीन पर आधारित कई कारखाने थे। कुछ कारखाने सल्फ्यूरिक एसिड यानी गंधक के अम्ल में को भी उपयोग में लाते थे, जिसकी वजह से हमें ऐसा लगा कि ये शायद त्रासदी घटी हो। अगले दिन हमने अखबार में पढ़ा कि वहाँ गैस रिसी थी, और बारिश हल्की होने के कारण वो गैस शायद पानी में घुलकर या बिजली चमक रही थी, उसके साथ रासायनिक प्रक्रिया द्वारा शायद तेजाब में बदल गई, और उसने ये अपना दुष्परिणाम छोड़ा।
अम्ल वर्षा के द्वारा भवनों में संक्षारण के कारण क्षति होती है। संगमरमर या लाल पत्थर की बनी ऐतिहासिक इमारतों और स्मारकों पर अम्ल वर्षा की छाप स्पष्ट रूप से देखी जा सकती है। आगरे के ताजमहल, मोती मस्जिद, लाल किला और फतेहपुर के अंबर महल को अम्ल वर्षा से काफी नुकसान हो रहा है।
जब कभी भी अम्ल वर्षा होती है, उसकी वजह से सल्फ्यूरिक एसिड इन पत्थरों के इस मुख्य अवयव यानी कि कैल्शियम कार्बोनेट से रासायनिक क्रिया कर लेता है, तथा कैल्शियम सल्फेट जैसे रासायनिक पदार्थ को बनाता है, जो स्पष्ट रूप से इन ताजमहल के सफेद पत्थरों को पीलेपन में बदल देते हैं। इसका एक दूसरा प्रभाव भी है। यही अम्ल जब संगमरमर के रासायनिक और भौतिक स्ट्रक्चर से क्रिया करता है तो उसको बदल देता है, और इसी बदलाव की वजह से उसमें दरारें भी पड़ जाती हैं।
अम्लीय वर्षा का विपरीत असर फसलों और भूमि पर भी हो सकता है। अम्ल वर्षा से भूमि में स्थित खनिज, पोषक तत्व और जीवाणु भी नष्ट हो सकते हैं। अम्लीय वर्षा से वन संपदा भी नष्ट हो सकती है। पेड़ पौधों के कुछ भागों का नष्ट हो जाना और इनके समुदाय के समुदाय नष्ट हो जाना अम्ल वृष्टि के परिणाम हो सकते हैं।
ऐसा जरूरी नहीं है कि जिन स्थानों से प्रदूषक गैसें निकलती हैं केवल वही अम्ल वर्षा हो। जर्मनी और यूनाइटेड किंगडम स्थित मिलों से निकलने वाली सल्फर डाइऑक्साइड और नाइट्रोजन के ऑक्साइड के कारण नॉर्वे और स्वीडन के विस्तृत क्षेत्र में अम्ल वर्षा होती है। इस वर्षा के कारण इन देशों की अधिकांश झीलों की जैविक संपदा समाप्त हो गई है। ऐसी झीलें मृत झील कहलाती हैं। अभी कुछ वर्ष पहले खाड़ी क्षेत्र में हुए युद्ध के दौरान कुवैत के प्राकृतिक तेलों के कुओं में आग लगी, जिससे उठने वाले धुएं का असर ईरान सहित कई अन्य देशों में भी देखने को मिला। कनाडा के ओंटारियो प्रांत से ओजेबिक तक की 2.5 लाख झीलों में से लगभग 50,000 झीलों में अम्ल वर्षा हुई है। इनमें से 140 झीलों को मृत घोषित कर दिया गया है। ऐसे ही अमेरिका के औद्योगिक क्षेत्रों से निकले ऑक्साइड का असर कनाडा और मैक्सिको में अम्ल वर्षा के रूप में होता है।
अम्ल वर्षा का मानव और पशु पक्षियों पर भी विपरीत प्रभाव पड़ता है।
इन एसिड की वजह से जो हमारे शरीर पे विपरीत प्रभाव होते हैं वे 2 प्रकार के होते हैं। एक इरिटेंट रिएक्शंस और एक कॉरोसिव रिएक्शंस। इरिटेंट रिएक्शंस एसिड के फ्यूम्स के वजह से होता है। उसमें मुख्यतः नाक से पानी आना, बहुत छींके आना, आँखों में जलन होना, आँख से पानी आना, या सांस लेने में दिक्कत आना, या अगर लेरिंजियल बॉक्स में सूजन आ जाए या श्वास नली में सूजन आ जाए तो उसके वजह से दम घुटने जैसा लगना। कॉरोसिव सिम्टम्स जो होते हैं वो जला देने की वजह से होते हैं। ये जितने भी एसिड्स होते हैं ये जिस पदार्थ के संपर्क में आते हैं उसे जला देते हैं। सबसे पहले हम सल्फ्यूरिक एसिड को लें तो इसकी पानी के प्रति बहुत अधिक लगाव होता है और इस वजह से ये जिस भी ऑर्गेनिक मैटर या टिश्यू या म्यूकस मेम्ब्रेन के कांटेक्ट में आता है तो उसमें से पानी सोख लेता है और बहुत अधिक ऊष्मा पैदा करता है, हीट पैदा करता है, जिसकी वजह से वो टिश्यू जलकर राख हो जाता है। तो यदि सल्फ्यूरिक एसिड की वजह से टिश्यू जलता है तो वो काला हो जाता है और इस रिएक्शन को चारिंग रिएक्शन कहते हैं। यदि नाइट्रिक एसिड की वजह से ऐसा होता है तो नाइट्रिक एसिड जो प्रोटीन्स होते हैं हमारे टिश्यूज़ में, स्किन में, उससे रिएक्शन करके पिक्रिक एसिड बनाता है जो कि पीले कलर का होता है और इसे हम जेंटोप्रोटिक रिएक्शन कहते हैं। तो नाइट्रिक एसिड की वजह से जो जलना होगा वो टिश्यू पीला दिखेगा, जला हुआ हिस्सा पीला दिखेगा, जबकि सल्फ्यूरिक एसिड की वजह से जला हुआ हिस्सा काला दिखेगा। अगर इन दोनों मिक्स हैं तो मिला जुला प्रभाव आपको मिलेगा।
अभी भी हमारे देश में अम्ल वर्षा की समस्या ने इतना विकराल रूप धारण नहीं किया है। वैज्ञानिक अध्ययन इस बात का संकेत दे रहे हैं कि अधिकांश भारतीय नगरों में वर्षा के जल में अम्लता का स्तर अभी भी सुरक्षा सीमा से कम है। लेकिन बड़े शहरों और औद्योगिक नगरों में वायुमंडल में सल्फर के स्तर में आश्चर्यजनक वृद्धि मापी गई है। इससे लगता है कि वो दिन दूर नहीं जब भारत में भी इस समस्या का विकराल रूप देखने को मिलेगा। यदि समय रहते इस पर ध्यान दें तो शायद हम अम्ल वर्षा के प्रकोप से बच सकते हैं। कारखानों से निकलने वाले धुएं की जांच और नियंत्रण इस दिशा में एक कारगर कदम हो सकता है। वाहनों के धुओं की जांच करके भी ये देख सकते हैं कि इनमें किस हद तक प्रदूषकों की मात्रा है। वाहनों के उचित रखरखाव और सही ईंधन के प्रयोग से वातावरण में इन जहरीली गैसों के प्रवेश को रोका जा सकता है। जहां पर वायुमंडल में सल्फर डाइऑक्साइड और नाइट्रोजन की सांद्रता अधिक नहीं है, वहां पर पेड़ पौधे भी इस दिशा में सहायक हो सकते हैं। सल्फर डाइऑक्साइड जैसी विषैली गैस को वृक्षों की पत्तियां तेजी से ग्रहण करती हैं। इसलिए सड़कों के किनारे, कारखानों के आसपास और अन्य जगहों पर वृक्षारोपण से भविष्य में अम्ल की वर्षा की संभावना क्षीण हो सकती है। अग्निहोत्र और हवन जैसे अनुष्ठान भी वायुमंडल के शुद्धिकरण में महत्वपूर्ण भूमिका निभा सकते हैं।
यदि हम चाहते हैं कि मानव सहित इस धरती के सभी जीव जंतु कायम रहें, तो इस पर्यावरण को शुद्ध और संतुलित बनाए रखना होगा। ये संपूर्ण विश्व के सामने आज एक चुनौती है। इसलिए जरूरत है कि पृथ्वी की संपूर्ण मानव जाति एकजुट होकर केवल अम्ल वर्षा से ही नहीं बल्कि अन्य प्रदूषकों से भी इस पर्यावरण को बचाए।
So welcome to this lecture series on microbiology. In order to study the unity, we must know how microbes originated. And microbes originated in the form of first life forms. The earth originated around 4.5 billion years ago, and it was from the sun, and after the earth was formed, it was a burning mass of gases. And gradually it cooled down, and as it cooled down, it liberated a lot of vapor, and actually that precipitated in the form of rain, and that cooled down the earth's surface. And at the next point, there was some microspheres created, which are nothing but specific areas referred to as microcosms. And these microcosms, you can see, are specific center or reaction center where life forms were formed. And these microcosms produced the first life form. And these life forms were in the form of a particular nucleic acid or the unit of life surrounded by cytoplasm and bound by a definite membrane. And the theory that explained this origin from inorganic life form to organic life form is known as abiogenesis. And after the first life form was created, from there they produced the complex life forms. This is called biogenesis. The origin of first life form is by the theory of abiogenesis. And this abiogenesis can be explained by the famous experiment by Urey and Miller. And as you can see in this slide, that there was a discharge tube where there was a mixture of methane, ammonia, water and hydrogen, and electric discharge was given with the help of two electrodes, and because of this electric discharge it created a complex jelly-like mass of protein. And this protein was considered to be the first form of life. The theory of biogenesis, which was actually proposed by Louis Pasteur, it stated that the first life form was a life, and then from this life form the other forms were created. It definitely doesn't explain how that first life was created. A particular cell is having a definite nucleus and there is a membrane. So this structural feature is present in all life form, and definitely these life forms are not visible to naked eye. Microbiology is considering all these microbes whose size is not more than 100 micron. What is 1 micron? It is 1000 part of 1 millimeter. So you can very well imagine that a particular bacterial cell which is even smaller than 100 micron is having different structure, where there is a membrane or there is a cell wall and there is a cytoplasm and all different organelles present within. They are in the order of even smaller than micron, that is angstrom or nanometer, which are smaller units. The microbes or the microbial forms, they all have a definite structure, and that structure is there is a outer membrane or an outer wall with a cytoplasm inside, there is a nucleus, there are different organelles, and this pattern is true for majority of the microbes. And when we come to the two major types of cell, we have two types. One is the eukaryotic cell and the other one is the prokaryotic cell. As you can see in this slide, this is a typical example of a eukaryotic cell, where there will be a cytoplasm and at the same time there will be nucleus, and what is important, there will be large number of membrane bound organelles present, and each of these membrane bound organelles being smaller than the nucleus, they are not really visible even under the microscope, but yet they play a very important role. Like say for example, a chloroplastid will help in photosynthesis, a mitochondria will help in respiration, an endoplasmic reticulum will bring about the compartmentation within the cell, but definitely these structures are not always present in a prokaryotic cell. This prokaryotic cell is a simple bacterial cell or a cyanobacterial cell, where first of all you can see there is no discrete nucleus. The genetic material is dispersed all along the cell and it is represented by the nucleic acid. But definitely there will be cell wall, there will be other different structures present as cytoplasmic projection. And these projections normally they are in the form of pili, in the form of flagella, in the form of fimbriae, and at the same time there is no discrete membrane bound mesosome is present, there is no distinct membrane bound mitochondria, because mitochondria is absent, there must be some other organelle which will carry out respiration. So its place is taken by mesosome.
So can you please explain about the difference between flagella and pili?
Yeah, you have to look at this particular figure. You can see that these smaller projections are pili and this big projection is flagella. So a flagella usually is much longer and a pili a smaller projection. A flagella will help in locomotion, but a pili will help in attachment. And particularly this attachment will be required when the bacterial cell is reproducing, that is sexual reproduction of the bacterial cell. And another important structural feature is there. Flagella is formed of a protein called flagellin, whereas pili is formed of protein called pilin. Okay, so you have understood the difference between a prokaryotic cell and a eukaryotic cell. They are structurally so similar, but yet they are so much different. Why different? Because of their structural hierarchy. You can very well see in the previous slide that so much complex structure all in a single cell, and here relatively simpler structure, but definitely in a cell which is almost 100 to 500 times smaller than the eukaryotic cell. These bacterial cells or these microbial cells, they follow some unique feature. That means they share some important similarity, and this similarity can be, first of all, structural similarity. Any microbe, they do have this membrane or a wall outside. They do have the locomotory organelle in the form of flagella. They do have pili or other projection. There will be nucleic acid. They share a common structural feature, which is the first important unity, first important feature to indicate that microbes have all originated from a simple cell having a nucleus inside. But because of these microbes, at least the prokaryotic microbes, they do not have a well-defined nucleus. They are called pro, meaning primitive, karyon, meaning nucleus. So they are called prokaryote. And this nucleus is dispersed within the cell without any definite membrane. Now coming to the next aspect, that is the metabolic pathways that they follow. The metabolism is the sum total of chemical reaction that takes place within this cell, and this metabolism can be actually catabolism or destructive process, anabolism or building up process. Now initially when we consider, you can see that this is the mitochondria, so this is the site of respiration. You will see that in all prokaryotic organism, mitochondria is absent, but its place is taken up by the mesosome, and the mesosome will also have the same purpose, that is generation of energy, and this generation of energy takes place through respiration. You know, respiration occurs mainly in two phases, and these two phases are glycolysis and Krebs cycle. And as you know, glycolysis is a process which occurs in absence of oxygen, where glucose molecule is converted to pyruvic acid, whereas Krebs cycle, which is conversion of citric acid produced from pyruvic acid via acetyl coenzyme A to a series of organic acid. But their purpose is same, that is generation of energy rich compound. And what are they? They are either ATP or NADPH2. So these compounds are produced in a living cell irrespective of the nature of this cell, that is whether they are prokaryote or eukaryote. And coming to the most important aspect, you can see this Krebs cycle is followed by all living organisms, and they are producing this NADPH, and this NADPH, which is reduced NADP or nicotinamide adenine dinucleotide phosphate, is taking place within the F1 particles of the mitochondria. But you will ask me that in bacteria there is no F1 particle, so it occurs within the membrane of the mesosome. But the purpose is same, that is this particular reduced NADP will be oxidized, and this oxidation process is not a very simple process. It will involve a series of electron carrier present across the membrane, and these electron carriers are mainly divided into at least 4 different complexes. What are they? The NADP complex, the coenzyme Q complex, the FES complex, and the cytochrome A3 or A complex. And ultimately, they will be generating the electron. A part of that electron will move out across the membrane, and as it moves out, it will activate a very complex structure. You can see this club-shaped structure, which is called ATP synthetase. And this club-shaped structure is activating the ATPase enzyme to produce ATP. So now you know that whether it is mesosome or whether it is mitochondria, their function is same, and that is energy generation in the form of ATP molecules. So now we start with the anabolic processes within the living cell of a microbe. The microbes, they produce fatty acid by the same enzyme fatty acid synthetase. The enzyme is same, but the location or the site of production may vary. In the same way, the microbes may produce polypeptide with the help of ribosome and with the same set of processes like the transcription and translation. So this particular anabolic processes will actually result in the increase in the volume of the microbial cell. Now coming to the code of life, which is the nucleic acid. From virus to all other microbes, they have the same pattern of genetic code, that is the arrangement of the nitrogen bases, and that is why the virus may be surviving on other microbial life, like bacteriophage surviving on bacteria. In this way, we can see that there is a circular plasmid present in a bacterial cell, and this plasmid can actually be transferred during the process of conjugation from one bacteria to the other, resulting in the modification of that particular bacteria. And this particular transfer is known as horizontal gene transfer. There may be some transmission where there will be one group of organism transmitting a particular gene product to higher organism. And this gene product, as you can see in a paramecium cell shown here, is through the production of kappa particle originally from a bacterial cell. This kappa particle is originating in the paramecium, and after it has been accepted in the cytoplasm, it is transferred via the cytoplasm. Whenever the conjugation process is very sharp, there will be no proper cytoplasmic exchange, so the kappa particle will not be distributed evenly, and they will result in production of killer and sensitive strain in an unequal manner. So this type of transmission is known as vertical transmission, and the genetic material ultimately will result in the modification of the microbe itself. As you can see in this particular phylogenetic tree, there is a single bacteria which is Pseudomonas, but because of this variation, they have created innumerable number of strain and species. And this is how one particular microbe originate as a single piece of microbe, but gradually it modifies and it will produce new species. When we consider the next topic, which is microbial ecology, you will see that they have the same purpose. Same purpose meaning they are sometimes acting as producer in an ecosystem, or sometimes they are acting as definite recycling agent. And this recycling is converting complex nitrogenous or other compounds to relatively simpler substances. And when we see that a particular bacteria is getting into this action of decomposition, we coin a very unique term called bioremediation. Bioremediation is conversion of some complex polluting substance to relatively simpler substances, like heavy metals are removed from nature, or like complex oil is degraded to relatively simpler hydrocarbon. And these microbes, when they perform this type of bioremedial activity, they will get into a different sort of cooperation. And this cooperation or interaction within microbes can be classified as positive and negative interaction. Positive interaction meaning when they are participating towards the benefit of each other. Like say for example, the algae and fungi together will remain in the lichen in an association called mutualism. And this type of association we normally find in protocooperation, that is the rhizosphere organism. But every association is not positive. there may be negative association, and this negative association can be parasitism. That means one particular parasite growing in an animal or growing in an animal microbe. It can be predation. This is a particular Daphnia, which is a zooplankton. It will predate upon different algal members, which are commonly known as phytoplankton. So this type of association always participate in nature. Between the same organism, always there is some sort of signaling, and this signaling takes place in microbe, and this signaling is called commonly the quorum sensing. Like you see in our dental plaque, there may be different bacteria colonizing and actually communicating with each other through chemical signals. So that also definitely indicate that microbes may be of various types, but they do exchange chemical signals between themselves. Microbes, in spite of their different nature, they can be controlled by a group of compound called antibiotics. A group of antibiotics which will commonly block protein synthesis is true for any other living organism. That means, say for example, all gram-negative bacteria may be treated by a single antibiotic, or there may be a group of vaccine created against a particular pox virus. So this is how we say the vaccine or antibiotics, they may be chemically different, but they all are used against specific group of bacteria or group of viruses. Now again we must understand that microbes can be of various nature, but they do have some similar pattern. And what is that pattern? They induce infection with the help of certain structure. We call it infection peg, or we call it pili. Whatever may be the cellular projection, their purpose is same, that is to infect. And at the same time, there will be some molecules like protein in the form of toxin, and this toxin will act upon these microbes, other microbes or other higher organisms, and will induce infection. And so we have seen that microbes started their journey as simple membrane bound cell, but they have produced various types. So here lies the diversity, and when we consider the diversity, we must consider what are the different types of microbes. Now the first group which I will show you is the group of algae, and these algae are unique photosynthetic microbes. There may be larger algae, but most of these algae are unicellular or colonial. As you can see here, we are representing a Volvox colony, and this Volvox colony have a unique pattern or aggregation of cell interconnected by cytoplasmic strand, which indicate that they are functionally having some connection. And at the same time, there may be small colonies within this large colony which represent daughter colony. So these organisms, they are producing a colonial group of microbes. And we also have mentioned about the bacteria. I told you these bacteria are unicellular prokaryotic organisms which do not have proper nucleus, but they may be of various types. They may be photosynthetic, they may be saprophytic, they may be parasitic, but all of them, they have a definite cellular pattern that is similar to any other prokaryote. Now coming to a group of microbes which represent this filamentous microscopic structure called mycelia. And this group of microbes are called fungi. And this fungi, as you know, will grow in the soil, will grow in dead and decaying plants and animals, or even can grow in living organisms, indicating infection. But these filamentous structures are interwoven, and they produce a compact structure sometimes in higher forms of fungi, which are commonly known as mushrooms. And these mushrooms, as you know, they may be consumed also. Mushrooms can be very much important for the diet because it has very little amount of fat, and at the same time it has high protein diet.
Sir, why we generally consume white mushrooms
than
the colored ones?
This is a good question. Actually, there is no test to determine whether that mushroom is edible or not. So the most important thing is to consume some mushroom which is white in color. Why? Now you see the mushrooms mostly grow in decaying wood or any cellular substrate where there will be lot of cellulose or pectin or lignin. Now whenever that particular mushroom will produce lot of cellulase or chitinase or pectinase, then all those complex cellulosic material will be totally digested or degraded, and thereby that poisonous component will go, so that white mushroom becomes totally edible. But if that mushroom is colored, what does it mean? Either it has been unable to digest cellulose or pectin totally, or it has produced some colored substance which may not be digestible by human or may not be tolerated by human. So they become inedible or they may become poisonous. Now another group of microbe which we will discuss is this protozoa. Now here you can see that we are seeing the Giardia, which is commonly known as the grand old man of microbial world. Now this protozoa will definitely have a membrane, will have a nucleus, and will have cilia. So protozoa commonly can move around and can induce infection, and very commonly the water of Kolkata city contain large number of amoeba or Giardia. So people living in this city may suffer from diseases which are chronic, causing amoebiasis or giardiasis, right?
Sir,
yes.
Sir, why Giardia is called as grand old man of microbe?
Yeah, you can see that this is a microscopic picture of Giardia, where there will be a single cell with a short neck and host of cilia. When you look it carefully, you will find that this is a man with 2 eyes, a bald head, and some beard. So it looks like a grand old man, so it is called a grand old man of microbes. But this particular Giardia is causing infection in the intestine, human small intestine, and it is a stubborn type of infection which will require long-term medication. So it looks like a grand old man, but it is very much harmful. Now we come to the next group, which is commonly known as helminth. Now you see, helminth, the group is represented by certain worms, and these worms are of 2 types, the flatworm and the roundworm. The flatworm, they are represented by the platyhelminthes, and the roundworm are represented by the aschelminthes. The most important feature present in these worms are that they are parasitic, they are present in the human intestine, so naturally they will not get any oxygen supply, and as because they are not getting any oxygen supply, they survive as anaerobic organism. As you can see here, there are examples of nematode or roundworm. You can see that there is a filamentous structure, and there will be a tubular structure which is their alimentary canal. So on one side there will be the mouth, on the other side there will be the reproductive pore. But definitely these microbes are sometimes very large. You can see the roundworm, which is commonly called the ascaris, it can be visible to the naked eye, but there may be very small microfilaria causing the infection of filariasis is not visible to the naked eye, and they are transmitted by some mosquito. You know that mosquito is Culex fatigans, which result in the spreading of this disease of filariasis. In other words, it is also called elephantiasis, because the person's leg is swelling like an elephant's foot, and it is because of the fact that all the lymphatic channels are blocked and the leg becomes very thick and fat. Okay, so these are the different helminth forms, and these infections are treated by the anthelmintic treatment or any compound that kill this particular infection. It is always suggested that because we suffer from mixed infection, on a regular basis we must go for this anthelmintic treatment so that our intestine is free from these microbial forms. We come to the last important form of microbe, which is in the form of these viruses. Now viruses are not true cellular organisms. They represent a group of organisms which is intermediate between living and non-living. Why living? Because these organisms are producing infection. They contain nucleic acid. They contain the same genetic code. But why non-living then? Because they don't have any cell. What are they having? They are only having an outer coat. You can see the outer coat and the nucleic acid inside. This is a bacteriophage. What is that? a virus that kills bacteria. And this bacteriophage is having a specific head and a long tail, and this tail is formed of helical protein, whereas the head is formed of capsid-like protein having specific geometrical structure. And you could see this particular head is represented by a 20-sided polygon, so we call it icosahedron. So a bacteriophage will commonly attack a particular bacterial cell, but it might kill it, it might not kill it. When it kills it, it is called the lysis of the cell, and the cycle is called the lytic cycle. But sometimes it will not kill it. The DNA or the genetic material will enter into the bacterial cell and will continue to replicate along with the bacterial DNA. And this cycle is called the lysogenic cycle. The last important aspect that we are going to consider is the way we name them, that is nomenclature of microbe. Like any other living organism, the nomenclature follows a specific epithet and a generic epithet. So we all follow a binomial nomenclature. And this binomial nomenclature is also having the author citation, that is the name of the author who reported it for the first time. So coming to Escherichia coli, we call it Escherichia, which is the generic name, coli being the specific name, and we all have some abbreviation. So the short name or the nickname is E. coli. So we can see that microbes started from a simple cell. They have developed certain commonality and they have produced different diversified forms, but yet they are all united because of certain typical similarity features, and they are also named in the same way. So students, we conclude this opening lecture, and what we have actually learned today, we have learned how microbes have originated in this world and how they have developed into structural complexity, yet how they are similar with respect to their cell structure, with respect to their genetic code, with respect to their exchange of genetic fragments, and of course with the respect of their infectability, interaction and control mechanism. At the same time, we have also seen that how microbes are performing together in the development and betterment of nature, and in what way actually microbes are playing a crucial role in the systemic formation of the nature. And we also have known the different types of microbes, starting from algae, fungi, bacteria, virus, parasites like protozoa, and also the nematodes or the helminths. And the last thing which we come to know from this lecture is the way we name them. I have told you that we have named them in a simple binomial nomenclature like any other living organism, with a generic name starting with a capital letter and a specific name starting with a small letter, and it is always in Latin like any other living organism.
Hello friends, very good morning and welcome to CEC live lectures. Yesterday I started with volatile oils, and in that we discussed about definition of volatile oils, then composition of volatile oils, what are the properties of volatile oils, how they are classified, and different examples we also discussed under different categories. So friends, today I am going to start with the extraction methods for the volatile oils, and as we discussed yesterday that extraction method, selecting an extraction method for extracting volatile oil is an important parameter that also has an impact on the yield of the oil as well as the quality of the oil. So there are different extraction methods today that are being used, but I'll be broadly categorizing those extraction methods into two categories. One is the traditional methods and the second one is the modern methods. Now, as we discussed that the traditional methods, mainly distillation is one of the oldest methods, perhaps the oldest method for extracting volatile oils, and distillation is of further three types that is largely used for extracting volatile oils, like hydrodistillation, that is water distillation, hydro steam distillation, or you can say water steam distillation, then third is the steam distillation. Now, apart from these three most commonly used distillation methods, there are other types of distillation, that is destructive distillation, which is normally carried in the absence of air, and vacuum distillation, which is I will say a bit modern method in comparison to these three distillation processes. Now, apart from distillation, as we discussed in the definition given by ISO, we also have an expression method. Again, expression methods will be further subdivided into traditional expression methods and the modern extraction methods. And in modern extraction methods, various mechanical methods have been developed. Obviously, these mechanical methods will also come under the expression methods, and these mechanical methods are sumatrous and pilatrous. These are the two widely used extraction methods for extracting volatile oils. Then we will have again one of the traditional methods, that is enfleurage, and after that I will be just giving a brief outline of the modern methods, specifically solvent extraction, then supercritical fluid extraction, what is solid phase micro extraction, head space trapping techniques, microwave assisted extraction, then we will have solvent free microwave assisted extraction, and molecular spinning band distillation apparatuses. So these are the extraction methods which we are going to discuss today. So starting with distillation, and as we discussed yesterday that it is the oldest technique to isolate the volatile oils, and this technique is still being used by industries or at laboratory scales. Now, distillation largely depend upon the insolubility of the volatile oils in water, and therefore it's a biphasic system is generated during the distillation. So as you can see in the picture, it is one of the primitive forms of distillation apparatus, where you can see one of the bigger container. It contains the plant material and water, and this bigger container is attached through the pipe to a smaller round bottom container, which is placed in a small tank at a level below the bigger container, and normally these tanks were filled with water, and whenever distillation was being processed in these containers, then the fumes they pass through these tubes into the collecting container, and since collecting container was placed in a water tank, these vapors get condensed, and because these vapors both contain volatile oil vapors as well as the vapors of water, and volatile oil being insoluble in water, two layers were formed in the collecting container, and from there the oil was separated. So this was the basic setup or the primitive setup of the distillation apparatus. Now, while you carry out distillation of volatile oils, there are three main physicochemical processes that occur during the distillation. First one is hydrodiffusion, second is the hydrolysis of the constituent, and third is the decomposition of those constituents by heat. Now, in hydrodiffusion, because we are boiling the water along with the plant drug, so water get absorbed through the plant cells, reaches to the localized cells where oil is present, and they cause rupturing of those cells. And since water is boiling, whenever the water comes in contact with the oil, it causes immediate evaporation of the oil, because oil is obviously a volatile oil, it will evaporate. During the hydrolysis process, when water comes in contact with the oil, there are certain constituents present in the oil which get hydrolyzed. Say for example, oil who are having esters in them, they get hydrolyzed into alcohols and acids. Since distillation is carried out at almost 100 degrees Celsius, because we are boiling the water, certain constituents also get decomposed by heat. So, all these three processes, they basically occur simultaneously in a very short span of time, and they have a great impact over each other. So, as you increase the temperature, the hydrodiffusion will increase. As the hydrodiffusion will increase, it will cause hydrolysis. As the hydrolysis occurs, then obviously since we are increasing the temperature, the decomposition of the components will also increase. Again, if you are increasing the time of distillation also, then again the diffusion process will increase, leading to hydrolysis and decomposition of various constituents. So, that is why I said that all these three processes, they go simultaneously and they have impact on each another. So, in order to obtain a better yield in terms of quality, then there are few precautions one can always take while distilling the oils from the plant. You have to maintain temperature as low as possible during the distillation. Now, the temperature should be enough so that the evaporation continuously takes place. So, we have to just maintain that temperature at which the evaporation continuously takes place, and then use as little water as possible. Now, the major function of the water is to rupture those cells and to pass into the pass through the membranes of the plant cells and rupture the oil cells and carry the vapors of the volatile oil towards the collecting container. So, the use as little as possible water as possible means that you have to use sufficient amount of water that just causes, or that is enough to submerge your drug material. So, there is no need to take excess of water while distilling the plant material. Only that much water is enough that will submerge your plant material. Then thoroughly comminute the plant material and pack it uniformly before distillation. And this is a very basic principle, because we know that as soon as we decrease the particle size of a plant material, the extraction efficiency obviously increases. So, this is nothing but we are decreasing the particle size, we are powdering the drug material and then packing it uniformly before the distillation. So, these steps basically help in improving the quality as well as the yield of the volatile oil. Now, coming on to the first distillation technique, that is water or the hydro distillation, and as we discussed, it is an oldest technique, and at laboratory scale Clevenger apparatus is largely used to distill the oils, and you can see the picture or the setup of Clevenger apparatus in picture A. You can see the round bottom flask is having the plant material and it is placed in a heating mantle, and to the round bottom flask is attached the Clevenger apparatus. Now, on the picture B, you will see the specifications of the Clevenger apparatus that are prescribed by WHO, and you can see in the picture that the two vertical tubes, one is the tube that is attached to round bottom flask, and the second tube is having the condenser and the collecting tube. Now, these two vertical tubes are attached to each other through an oblique tube in the center, which is having a cup shaped apparatus attached to it. Now, when the distillation starts, the water starts boiling, it will carry the vapors of the oil along with the water vapors. They will pass through the tube A, first vertical tube, and they will move in the upside direction, and through the upper connecting tube they will enter into the condenser, where both the vapors of oil and water, they will condense and they start getting collected in the collecting tube. Now, the collecting tube and this oblique tube, which is connecting both the vertical tubes, is filled with water. Now, you can see in the picture A that this is a complete closed system. So if water evaporates from RBF, you cannot add water to it. So if you want to add water, then you have to stop the distillation, you have to remove the Clevenger apparatus. So in order to have a continuous distillation process, then this oblique tube role comes into play. And since I told you that this tube is filled with water, so as the water evaporates from RBF gets condensed, so the amount of the water that condensed from the condenser, it enters into the collecting tube, and the same amount of water through this oblique tube enters back into the RBF. So I will explain this process, what this process is known as, in the subsequent slides. Now coming back, Clevenger is largely used, or this hydrodistillation is largely used to prepare ether. Ether were the perfumes that were used in yesteryears by so many years back, and they were largely used for cosmetic purpose only. Now the major advantages of water distillation is that it's a very inexpensive technique, both in terms of the apparatus used as well as the solvent. Apparatus you have seen, it's the Clevenger apparatus, or if you are using this water distillation at a larger scale, then also you will be just needing two vessels, one larger one and one smaller one. So the overall cost of setting up a distillation apparatus is less in comparison to other techniques. Vis-a-vis, the solvent which we use is water, and water is the cheapest and the safest solvent known today, and that's why it is considered as an inexpensive technique. Now during the distillation, water continuously is boiling, and that is why it helps in continuous motion of the plant material, which prevents the formation of the lumps during the distillation, and that is why the distillation is having a good efficiency. I will say not very good in comparison to the other methods, but yes, because lumps are not formed, because water is continuously boiling, which keeps on moving the plant material, thereby preventing the formation of lumps. That is why the extraction efficiency is improved. Now coming on to the disadvantages, now complete extraction is not possible. When I say complete extraction is not possible, there are certain constituents which require higher boiling points, more than 100 degrees, and those constituents do not get distilled out when you are using water distillation, because you have to boil the water and you cannot go beyond 100 degrees in water distillation. And again, you have to keep the temperature low, that is below 100 degrees, in order to get a good quality oil. So sometimes the extraction is complete extraction is not possible. Second major disadvantage is that this process is not suitable for thermolabile constituents, because these type of constituents get degraded during the distillation process. Like certain esters are partially hydrolyzed, and sensitive substances like aldehydes, they tend to polymerize whenever the distillation is being carried out, because you are heating the mixture continuously. Then another disadvantage, the high boiling and somewhat water soluble constituents like phenols cannot be completely vaporized or require prolonged extraction, thus sometimes become uneconomical. This is what I was discussing when I said that sometimes complete extraction by this process is not possible. This is only because the high boiling compounds or the water soluble compounds, they do not get completely distilled off. Therefore, the complete extraction is not possible, and you have to go for prolonged extraction, which further increases the risk of degradation of the components, and sometimes the whole process becomes uneconomical. Next we have is the water and steam distillation. This is a slight modification of the hydrodistillation, and in the picture you can see that the plant material is placed on a mesh, and below the mesh is present the water, which is continuously heated, and as we boil the water, the steam rises and it passes through the mesh and it comes in contact with the plant material. And since steam is having higher latent heat than water, it immediately ruptures those oil cells present in the plant parts and liberates the volatile oil, which get evaporated, and the vapors of water in the form of steam as well as the vapors of the oil, they move upwards, and through that red tube they enter into the condenser, where both the vapors get condensed, and then the condensate is collected in a collecting vessel, and since oils are insoluble in water and most of the oils are lighter than water, they form an upper layer, and from there you can easily collect the oil and you can easily separate the water from it. So this is just a modification. Here we are not boiling the drug with water, we are just extracting the oil with the help of steam that is generated by the water within the same container. Now the major advantage of this technique is it provides a better quality oil, because the plant material is not in direct contact with water. So therefore the hydrolysis aspect or hydrolysis part of the water distillation is being taken care of in this water and steam distillation. Cohobation is another process which can be attached to the water and steam distillation as well as the hydro distillation. Though this process can be used in all the three types of distillation, but cohobation is widely used in water and steam distillation process as well as in the water or hydro distillation. Now cohobation is nothing but the recirculation of the distillate water, and as you can see in this picture, this is a pictorial representation of water distillation or the hydro distillation. You can see that plant material is being boiled in the water, and with continuous stirrer has been attached to it. The vapors of the oil and water, they are passing through the white tube and enter into the condenser where they get condensed, and as you can see in the collecting chamber or the collecting container, oil being lighter than water is forming the upper layer from where it is separated, and the water through a cohobation system or the cohobation pump is being recirculated back into the distillation container. So if we go back into the Clevenger structure or the Clevenger apparatus structure, you can see there is an oblique tube which I told you. This oblique tube helps in cohobation process. So whatever water condenses in the collecting tube, same amount of water through this tube passes back into the RBF or the distillation container. So this is all about the cohobation process. Now coming on to the other advantages of water steam distillation process, one is as I told you, better quality oil is obtained because the hydrolysis is less in this type of extraction method. Along with this, since this process is carried out with the help of steam, higher yield of the oil is obtained. Second point is the components, again they are less susceptible to hydrolysis and polymerization because we are using steam, you are not directly boiling the plant material into water. And the third point states that steam and water distillation is faster than the water distillation, so it is more energy efficient because steam is having more latent heat in comparison to water. Therefore the overall distillation process is much faster in comparison to the simple hydro distillation or water distillation, and therefore this process becomes more energy efficient. Now coming back to the disadvantages of this process, now we are just boiling the water and the steam is rising which passes through the plant material. Therefore that steam which passes through that plant material has a very low pressure, and therefore the oils or the oils having high boiling range constituents, they require greater quantity of the steam for vaporization and hence longer duration of the distillation, which obviously sometimes becomes uneconomical. And during the steam distillation, sometimes plant material also gets wet because as soon as the steam comes in contact with the plant material, some of the steam get condensed and the plant material becomes wet. And once the plant material becomes wet, the overall distillation process sometimes get slow because first now the steam has to first evaporate that water from the plant material and then it will evaporate the oil. So overall process sometimes becomes slow if the steam gets condensed or the plant material gets wet during this process. Now the third type of distillation is the steam distillation, and as you can see the setup of the steam distillation, you can clearly see the difference between the steam distillation and water and steam distillation. Here the steam is being generated outside the distillation container, and once the steam is generated, this steam is then passed into the distillation container where plant material is placed on a mesh. Now this steam passes through again through the plant material. Rest of the process is quite similar as that of the water and steam distillation, but the only difference is the steam is being processed or steam is being generated in a steam generated outside the distillation container. Now, what are the advantages of steam distillation? The amount of steam, since it is being generated outside the distillation container in a separate container, one can easily control the amount of the steam as well as the pressure of the steam by which you have to pass that steam through the plant material. So, this is one of the biggest advantage of only steam distillation. Now, since steam is generated in a boiler, the plant material is heated at a temperature not more than 100 degree and consequently less thermal degradation is there. This also happens in the water and steam distillations. And this process is the most widely acceptable process by the industry for the large scale production, oil production. Since the steam distillation process can have a very good scale up method, so you can easily set up an equipment or a system having larger quantities, and therefore this method is widely acceptable and a widely accepted process by the industry for the large scale oil production. Now, coming on to the disadvantages of only steam distillation, since you can go for a scale up of this method, much higher capital expenditure is required, which makes this overall process somewhat uneconomical. And sometimes if you are extracting out low cost oils with this technique, then also this technique sometimes become uneconomical. But nowadays industries are using this technique widely to extract even some low cost oils, to overcome the labor orientation, to overcome the time duration or the time which is involved in other types of distillation. So, to overcome they are start using this process. Now, the next type of distillation is the destructive distillation, which is used in some special cases, and as I told you that it is carried out in absence of air. It is mainly done to extract oil from wood or the resins, and the wood or resins, when heated in the absence of air, the decomposition of the tissues takes place and number of volatile compounds are driven off, or you can say distilled off. And whatever volatile oil, volatile matter is condensed, it is separated into two layers: an aqueous layer containing wood naphtha, which is generally methyl alcohol, and pyrogenous acid, which is a crude acetic acid. This is one layer, and the second layer is of a tarry liquid, which is in the form of pine tar or juniper tar and other tars, depending upon the plant material or the wood you are using. So, if you are using a pine wood, then it will form a pine tar. If you are using a juniper wood, then it will form a juniper tar. So, depending upon the type of the plant material you are using, you will get a similar kind of the tarry liquid. Next we have is the vacuum distillation, and it is generally used for extracting volatile components having higher boiling points, but can also be used for lower boiling point volatiles. Since you are applying the vacuum basically decreases the boiling point of the solvent or the oil, which further increases the extraction efficiency as well as the quality of the oil, because you are carrying out now the distillation under vacuum and at a very low temperature. Therefore, it also avoids the thermal degradation. Next we have is an expression method, or you can also call it as cold expression method. This was all about distillation. Now we move on to the expression method, and as I told you that these expression methods are also categorized into two broader categories, traditional methods and the modern methods. Now, the traditional, there were two methods, one is sponge method and second is Scodella method. In sponge method, the peels of the citrus fruits were separated by a knife known as rastrello. Now, before we move further, these expression methods are largely used to extract oils from the citrus fruits. So, once these peels are separated from the citrus fruits, they are rubbed on a clay brick, and they are continuously washed with water. Now, this oil and water mixture is being absorbed on a piece of sponge. Because you are continuously washing the rubbed material, it will pass on wash down to a piece of sponge where it get absorbed. So, once the sponge piece gets saturated with the oil and water mixture, it is squeezed and that mixture is separated from the sponge, and that mixture is allowed to stand for some time, where it causes separation of oil and water, and from there you can separate the oil. The only disadvantage of this method is that it's quite time consuming. Than the oil obtained by this method is having more of a fruity odor rather than its natural fragrance. Next traditional method is the Scodella method, and this was slight advancement of the sponge method. Here we take a bowl having raised blunt ends, and this bowl is attached with a funnel at the bottom. So the peels of the citrus fruits are rubbed on these blunt ends and is continuously washed. So the water and oil mixture washes down through the funnel into the collecting container, and where the oil is separated from the water. Again, the disadvantage is it's a very slow and manual method and labor oriented method, having a very low yield. Then further modifications were done in the expression method, and the modern method is the Éculée method. Though Éculée also is, I will not say it's a recent method, but in comparison to sponge and Scodella methods, it's a new method. Now here we again, you can see we have a funnel like apparatus having pointed projections on it, and it is attached through a hollow tube below. So the peels of the citrus fruits are rubbed on these pointed projections, thereby rupturing the oil cells or the oil glands that are present in the peels, and it is continuously washed with water, and the water oil mixture passes down through this hollow tube into the collecting container, where the oil is separated from the water. Now this Éculée's method or these expression methods have been mechanized, and one of the method is Pilatres method, where the peels of the citrus fruits are feeded through a hopper, have an Archimedean screw inside, you can see in the picture, and the walls where the screw is rotated, they have abrasions on the surface. So when the peel comes in contact with these Archimedean screws, they start rotating, and this causes rubbing of the peels on the abrasive walls, which cause rupturing of the oil glands, and the oil is continuously being washed off these screws, and the washings, water and oil is collected below, from where the water is separated. And the second mechanical method which is widely used is Sfumatres method, and this equipment consists of a metallic chain that is drawn by two horizontal ribbed rollers, and these rollers lead to the pressing rollers which have abrasions on their wall, and these abrasions again they basically cause the rupturing of the oil glands, thereby releasing the oils, which is continuously washed with the help of water into the collecting container, from where the water is separated from the oil and oil is separated. So friends, I'll stop here, and in the next lecture I'll continue with the enfleurage method. Thank you. So welcome back friends, as we were discussing the extraction methods for volatile oils. So next in the series is the enfleurage method, and again this method is one of the oldest technique that is being utilized to prepare concretes and absolutes. Further, it is also used to extract oil from certain delicate flowers or the delicate plant parts like petals. Now this technique is largely used for for extracting those oils which have high percentage of thermolabile constituents, because you cannot extract these type of thermolabile constituents or the oil having high percentage of thermolabile constituent using distillation methods. Now, the major advantage of enfleurage method is it retains the original composition vis-a-vis original odor of the natural odor of the oil, because you are not exposing the plant material towards the heat. Therefore, most of the components like ethereal components or aldehydes, they do not get modified, they do not get hydrolyzed, they do not get polymerized. So, you get more or less a natural composition or the original composition of the oil, vis-a-vis since no heating is there, you get the natural odor of that particular volatile oil. Now, enfleurage is of two types, cold enfleurage and the hot enfleurage. Now, the cold enfleurage is normally carried out at room temperature, that is why it is known as cold enfleurage. Here the petals or the flowers, they are pressed on a layer of fat which is spread on a glass plate, and this glass plate is fixed in a wooden frame. As you can see in the picture, picture 1 shows the enfleurage process, where these flowers or the petals are fixed on the fatty layer. Now, once these flowers or the petals are fixed on the fatty layer, the volatile components, they get absorbed in the fat layer and get exhausted. Once they are exhausted, they are carefully removed, and again this process is manual, and this removal of the exhausted flowers or the petals is known as defleurage, which you can see in the second picture, where a lady is removing all those flowers from or the petals from the fatty layer. Now, once these flowers or petals are removed, they are replaced by the fresh flowers or the petals, and the process is continued till the fatty layer gets saturated with the oil or the volatile constituents. Now, once the the fatty layer is saturated with the oil or volatile constituents, the fatty layer is removed, and the fatty layer is then extracted with the ethyl alcohol. So, I will go back to yesterday's lecture, where I discussed that one of the term pomade is largely used in connection with the volatile oil or essential oils, and I told you that the fatty material, that is the saturated fatty material that is obtained during the enfleurage process, is called pomade. So, here I will come back to enfleurage. So, once the fatty layer gets saturated with the volatile oil or the volatile constituent, we call it as pomade, and this pomade is then subjected to the extraction with organic solvent, non-polar organic solvent such as hexane or petroleum ether or toluene, and the distillate or the extract which we obtain is called as concrete. Again, if we are extracting this pomade with the help of ethyl alcohol, then the product which we obtain is called an absolute. So, these are the two products which you can obtain through the enfleurage process. Now, this second type of enfleurage is hot enfleurage, and this process is also known as maceration. Now, in this petals are normally they are dipped in a melted fat. This is a difference from the cold enfleurage, where you are fixing the petals and flower on the solid fat. Here you are dipping all those petals or flowers in a melted fat, and they are continuously heated for 1 hour at a temperature of 45 to 60 degrees Celsius. Now, again the volatile constituent or the volatile oil, they get absorbed into the melted fat, and once the plant material is exhausted or the fatty layer or the melted fat is saturated with the constituent, the fat is filtered off from the petals and the flowers, and the melted fat is then extracted with the alcohol to get absolute. Now, the hot maceration process is largely used for the flowers which have a very short shelf life after plucking, like lily of the valley, which is commonly known as Convallaria majalis. So, these flowers have a very short shelf life, therefore, they are the oil from these flowers normally extracted with the help of hot maceration or the hot enfleurage process. Now, the next process we have is the solvent extraction, and it is largely done using the volatile solvents, mainly non-polar solvents such as petroleum ether and hexane. And the major advantage of solvent extraction technique is the extraction can be done at uniform temperature, usually 50 degrees Celsius, and can be maintained throughout the process. And since the extraction is done at a very low temperature, thermal degradation is minimized in this process vis-a-vis the oil that is obtained, it retains its natural odor, and very less alteration in the chemical composition of the oil occurs, because you are heating or you are extracting the oil at a very low temperature. Now these are the extraction methods. Now I move on to the modern methods like supercritical fluid extraction, where we use supercritical fluids such as carbon dioxide. Now when carbon dioxide is a gas, but if you expose carbon dioxide at a temperature and pressure beyond its critical value, then the carbon dioxide will act as a solvent. And since the conditions or the critical conditions for the carbon dioxide can be easily attained, so carbon dioxide is the most widely used solvent in supercritical fluid extraction. Now again you are not exposing the plant material at higher temperature or pressure in supercritical fluid extraction, therefore the yield of the oil is improved, vis-a-vis quality of the oil is improved. Another modern extraction method is solid phase micro extraction, where you have to extract very small quantities of the oil from the plant material. In this, what we do is we take a polymer or an adsorbent in a small tube, and this tube is attached to a bigger tube through which a warm air is being passed, and this tube is placed over the plant material, and the plant material is heated. The volatile constituent or the oil get evaporated, and it gets adsorbed on that adsorbent present or the polymer present in the tube, and from there it is extracted with the help of solvent. And nowadays microwave assisted extraction is also being used to extract volatile oil, and further the plant material is heated using the microwaves, thereby decreasing the overall extraction time or the distillation time, because you can distill the oils using microwave also. So the overall extraction time is reduced in the microwave type of extraction. Then microwaves are also used for a solvent free extraction, where the plant material is heated without any solvent. This helps in reducing the traces of the solvent in the volatile oil. But the major disadvantage of these techniques, microwave or solvent free microwave assisted techniques, is only the thermal degradation of the constituents of the volatile oil. So this was in brief about the modern methods that are being used today for extracting volatile oils. Now we move on to the uses of the volatile oils. Again the uses for the plants, because as we discussed yesterday that these are the defensive chemicals or defensive agents for the plant itself. So whenever they are attacked by a microorganism or they have any pest infestation, they start producing these chemicals as a defensive chemicals. Then these volatile oils also act as attractants for various insects which help in their pollination. And since volatile oils have a very good antimicrobial properties, they protect the plants from various bacterial or fungal infections. Now apart from the uses in the plants, volatile oils have great uses for man, and they are widely used in perfumery industry, though today most of the perfumes or the constituents that are being used in perfumery are the synthetic ones, but still some people do prefer the natural oils that are being used to prepare these perfumes. Next major use is in aromatherapy, and I'm sure everybody knows about aromatherapy, where we treat certain diseases with the help of the aroma of these natural oils. And aromatherapy has been further categorized into aromacology, where the odor of these oils is used to attain the overall the psychological well-being of a person. So it is more or less the relation between odor and the psychology of the man, that is aromacology. And next is the aromatology. It is nothing but when the oil are administered inside the body through oral route or anal route or the vaginal route. Now many believe that since once the oil is administered in the body, then the role of aroma is not there, and therefore aromatology is not being considered as a part of aromatherapy. Now the modern aromatherapy was given by René Maurice Gattefossé, who is known as the father of aromatherapy. Basically, he was having a perfumery of his own, and one day while working in the lab he burned his hand, and immediately he dipped his burnt hand in a container having lavender oil, and he was amazed to see the therapeutic benefits of lavender oil on his burnt skin, and that's why he start exploring the therapeutic uses of the oil or the aroma of the oil, and hence he gave birth to the term that is aromatherapy. According to him, aromatherapy has been categorized into cosmetic aromatherapy, which is largely concerned with the nourishment of the skin or hair and the anti-aging processes. Then massage aromatherapy, which is largely related with the strengthening and relaxing of the muscles, and therapeutic aromatherapy, which largely deals with the treatment of specific diseases. Now, in aromatherapy, the oils are normally used topically through the skin, or they are used as inhalations, they are used in baths and saunas. Now, coming on to cosmetic aromatherapy, as I told you, it is largely used for the nourishment of skin and hair. Various types of oils like lemon oil, orange, rose, sage, tea tree oils, they are largely used in cosmetic aromatherapy. Then we have massage aromatherapy. Moray in 1989 gave the concept of massage aromatherapy, where she tried to explore the benefits of the oil when they are absorbed through the dermal layer of our skin. And various oils like orange, coriander, eucalyptus, mentha, juniper, geranium, and so on and so forth, they are used for the massage therapy in order to strengthening of the muscles or relaxing of the muscles. And in therapeutic aromatherapy, as I told you, that it is largely used to treat specific disorders like insomnia. We use valerian oil, lavender oil, lemon or Malacia oil, and to treat anxiety and stress we use angelica oil, orange, patchouli, valerian and geranium oil. To treat cognitive disorders or to improve cognition we use rosemary, mei-chang oil, sage oil or peppermint oil, and to treat the pains in bone and joints we use cinnamon oil or ginger, rosemary or chamomile oil. Now, apart from this aromatherapy, which is more or less an alternative system of medicine or a complementary system of medicine, the essential oils are also used for their therapeutic benefits, such as some of the oils like fennel and coriander. They are used as carminative, and I am sure everybody must have tasted the fennel oil when they were infants or babies, because babies or infants normally they have colic pain, flatulence in their stomach, and to reduce that colic pain or flatulence, fennel oil is normally administered orally. Then some of the oils are also used as local analgesic. The best example you can remember is clove oil, which is widely used as dental analgesic to treat toothache. Then antispasmodic like bitter almond oil. Juniper oil is used as a diuretic. For insomnia or as sedative, valerian oil is used. Eucalyptus oil is having very good antiseptic properties, and peppermint oil is more or less used as a stimulant. Now, apart from these medicinal uses, most of the oils have antimicrobial actions, and they have a very good bacteriostatic or fungistatic or virostatic action. They basically increase the permeability of the membrane of the microorganism, which causes loss of ions and reduction in the membrane potential, thereby causing the death of the microorganism. Then they also causes the collapse of the proton pump in the microorganism, thereby decreasing the production as well as the pool of the ATP, which further causes the death of the microorganism. Now, apart from these mechanisms, they also cause coagulation of the cytoplasm. They damage the lipids and the proteins that are present in the membrane, thereby causing the lysis of the microorganism or the bacterial cell. They also cause mutagenicity in the nucleus and the cytoplasm of the microorganism, thereby causing its death. So, all these mechanisms, they are shown by the volatile oils. By virtue of these mechanisms, they have exhibited very good, very potent, I will say, antimicrobial action. Now, these were the medicinal uses. Now, we move on to the pharmaceutical uses of these volatile oils, and in pharmaceutical industry, they are largely used as flavoring agents, odor masking agents, and as preservatives because of their strong antimicrobial action. Now we move on to another important aspect, that is analysis of volatile oil, because since they are applied topically, they are administered, they are used in pharmaceuticals, so the quality of the volatile oils become a great challenge, because normally these oils, the original or the natural oils, are very expensive, and they have a tendency to get adulterated, and the type of adulteration that can be done in volatile oils can further be categorized into simple adulteration, and the second one is the refined adulteration or sophistication. So in simple adulteration, the addition of natural or the synthetic compounds with an aim of generating an oil characterized by specific quality values such as density, optical rotation, residue percentage, ester value, and so on and so forth. Now in this simple adulteration, what is being done is sometimes synthetic or some other natural compounds are added in order to have the same or the physicochemical parameters value in the prescribed range, and that is why it can be easily detected. Whereas refined sophistications, these involve the reconstitution and counterfeiting of commercially valuable oils, and in these the natural and synthetic compounds are added to enhance the market value of that particular oil, and the adulteration done is in such a manner that it makes adulteration or the detection of the adulteration a very troublesome task, and it's very difficult to detect such kind of adulteration. But still there are certain parameters which can be used to detect the adulteration. One is specific gravity, which is nothing but the ratios of the density of the oil to water at a particular temperature, and the common range of the specific gravity of different oils fall in the range of 0.696 to 1.118 at 15 degrees Celsius. Now if you have to change the temperature, then you have to add the conversion factor if you are determining the specific gravities at different temperatures. So if your sample is having a specific gravity beyond or below the prescribed limits, one can easily say that it has been adulterated, or it is of poor quality. Next is the optical rotation, and it is measured with the help of polarimeter. It is angle of the rotation of the plane polarized light at a particular wavelength and temperature, and since most of the constituents in the volatile are optically active and they are present in the isomeric forms, optical rotation becomes an important parameter for assessing the quality or the assessment of the purity of the volatile oils. Next we have is the refractive index, and it is the ratio of the angle of incidence to the angle of refraction of a beam of light that is passing from a less dense to a denser media, and Abbe's refractometer is the major equipment or the most widely used equipment, which is normally equipped with the monochromatic sodium light source. Suppose if you are measuring the refractive index at a lower or a higher temperature from 20 degrees, because normally it is being done at 20 degrees or at room temperature. So if you are varying a temperature at which it is being determined, again you have to add or subtract the correction factor accordingly as per degrees. Apart from these physical methods, there are certain solubility tests which can also give you an idea about the quality of the oil. So for the oils which are rich in phenolic compounds, what you can do is you can take small amount of oil and dissolve it in aqueous potassium hydroxide solution, and if the solubility of oil is decreased in comparison to the standard oil, it will indicate the presence of impurities. Same if the oils are rich in aldehyde, then you can dissolve the oil in the solution of dilute bisulfite, then incomplete dissolution will indicate the impurities. And if your oil is rich in ketones, then you can use neutral sulfide in place of dilute bisulfide, and again the incomplete dissolution will show you the presence of impurities in that particular oil. Now one can easily detect the presence of water in the oil also. So what happens is, normally the oils that are rich in oxygenated compounds or phenols, they have tendency to absorb certain amount of moisture or water. So in order to detect the presence of water, the oil is dissolved in an organic solvent, say chloroform or hexane or petroleum ether, and then small amount of water if present in the oil, then the mixture will become turbid. So if the oil is having small amount of moisture, when you will add an organic solvent, so you will get a turbid mixture, whereas if the oil is free from moisture, then the solution or the mixture with an organic solvent will be absolutely clear. Now, apart from these traditional methods, there are certain modern chromatographic methods that are also being utilized to analyze these volatile oils, and these are thin layer chromatography, where we develop fingerprint TLC profiles of various oils, in which we determine the number of spots, the color of the spots, their RF values, the intensity of the spots, and then we compare that fingerprint profile with the profile of a standard oil, and if the test oil is having almost similar kind of profile in terms of the number of spots or the intensity or the color or the RF, one can easily get an idea about the quality of the oil. Then we can also use HPTLC profiles to determine, to assess the quality of the oils in the similar manner, and we can also quantify a particular constituent of the oil using HPTLC or TLC densitometric analysis. Same in HPLC also, we use HPLC chromatograms, and we can develop fingerprint chromatograms on HPLC, and we can also quantify a particular constituent present in the oil. And last and the most widely used chromatographic techniques for the volatile oils is gas chromatography, and with gas chromatography we can use various detectors like flame ionization, thermal conductivity detectors, or mass spectrophotometers. And amongst these three, GCMS along with MS, that is mass spectrometer, it is the most widely used equipment for assessing the quality of the volatile oils. Now we move on to the safety and toxicity concerns of these volatile oils, because we have now covered the therapeutic uses or the aromatherapeutic uses of the oils. Apart from these uses, these oils have certain safety concerns also, and the number one is the allergic reactions which these oils can cause, because some of the oils, they are rich in certain type of constituents like benzyl alcohol or cinnamyl alcohol, eugenol, hydroxycitronilol, or isoeugenol, benzyl salicylate, or cinnamaldehyde, and so on and so forth. And whenever these oils are applied on the skin, if a person is allergic to these constituent, he will immediately show allergic reactions of the skin, which can be assessed in terms of reddening of the skin, or rashes on the skin, or the itchiness of the skin. So allergic reactions by these oils is one of the major concern. Next concern is of carcinogenicity. Some of the volatile oils have shown carcinogenic action, and I have taken example of the oil of Salvia scaleria and Melaleuca quinquenervia, which provokes the secretions of estrogen, and they have shown to induce the estrogen dependent cancers in various in vitro studies, though these types of studies have to be corroborated by the in vivo results, but in vitro studies they have shown to induce the estrogen dependent cancers. Likewise, oil of Citrus bergamia can induce the skin cancers, as it has been shown in different studies. Next safety concern or the toxicity concern is their phototoxicity or photosensitization. Certain oils, they are rich in coumarin type of compounds or soralanes, where they absorb the sunlight and causes phototoxicity, and this phototoxicity is in terms of hyperpigmentation of the skin. And as you can see in the picture, on the cheeks or on the hands, the person who was allergic or who exhibited the toxic effects of the oil of Citrus bergamia, and you can clearly see the hyperpigmentation on the hand. So these are the few safety or the toxicity concerns of these volatile oils, and what can be done to prevent these toxicity effects. I will discuss in next slide. There are certain other effects like migraines, headache, dizziness, nausea, shortness of breath, or asthma that are also caused by certain volatile oils. Now, what precautions one can do is that you should always dilute your oil before use, and for diluting, you can always use certain carrier oils, and this dilution and the use of carrier oils has been seen in aromatherapy, and carrier oils like coconut oil or olive oil are largely used before these volatile oils are applied on the skin. Now, one can also take a precaution that these volatile oils are free from the traces of the solvent that has been used to extract these volatile oils, specifically if organic solvents are used, because it has been observed certain organic solvents have carcinogenic effect. And third, before using these volatile oils, one can always perform a skin patch test in order to see whether the person who is going to use it, whether he or she is allergic to these oils or not. So by following all these precautionary steps, one can always keep these allergic reactions or the phototoxicity reactions or carcinogenicity at a bay, and one can easily use these oils for cosmetic, therapeutic, massage purposes. So friends, this was all about the volatile oils, starting from their definition, their properties, their localization in the plant parts, their classification, their composition, biosynthesis, then various classes of the volatile oils, then various methods of their extraction, moving on to uses, their analysis and safety concerns. Thank you.
Hello viewers, in our previous lecture on molecules of life, we had discussed the elements and functional groups present in the biomolecules. As already discussed, living organisms, whether they are microorganisms, plants or animals, are basically made up of the same chemical components that are carbohydrates, proteins, lipids and nucleic acids. Since we are discussing about the molecules of life, we have to appreciate biochemistry as the study of the way in which these components are synthesized and utilized by the organisms in their life processes. That means we have to discuss the mechanism of life processes. Biochemistry bridges the gap between the conventional chemistry and biology. Each life process is explained through its chemical rationale, and the role of this single process is interconnected to the general metabolic pool of living organisms. In other words, life is nothing but thousands of ordered chemical reactions. One can therefore claim that chemistry is the logical explanation of all biological phenomena. Let me first introduce you to some of the pioneers of biochemistry and their contributions, which has led to advancement of our insight into life processes. Let us start with Carl Alexander Newberg. He was pioneer of biochemical studies. He is often referred to as father of modern biochemistry. And fructose 6-phosphate is named after him and called as Newberg's ester. Newberg introduced the term biochemistry in 1903. The discipline has grown as one of the most important subjects explaining life and its functioning. The real push to biochemistry was given in 1828 when total synthesis of urea from lead cyanate and ammonia was achieved by Wohler, who thus initiated the synthesis of organic compounds from inorganic compounds. In fact, Wohler was trying to prepare ammonium cyanate by reaction between lead cyanate and aqueous ammonia. And in the process he got ammonium cyanate, but this ammonium cyanate so prepared decomposed to ammonia and cyanic acid, which reacted reversibly to produce urea. That in conclusion we can say that when lead cyanate was treated with aqueous ammonia, ammonium cyanate was formed, but ammonium cyanate isomerized to form urea. As you can see on your screens, the molecular formula of ammonium cyanate and urea is same. And this was a vital blow, or I can say this, to the vital force theory. He was not trying to prepare urea in the lab, he was trying to prepare ammonium cyanate. And the ammonium cyanate formed decomposed to give urea. And then Wohler reportedly wrote to his teacher Berzelius, who was a staunch believer of vital force theory. According to Berzelius, organic compounds could be synthesized only in nature under some mysterious force. And then Wohler wrote to his teacher Berzelius, and I quote, "I cannot, so to say, hold my chemical water and must tell you that I can make urea without thereby needing to have kidneys or anyhow an animal, be it human or dog. In summary, he said that a biomolecule, that is urea, could now be prepared in vitro. Till now it was believed that urea can only be prepared in living organisms, in kidneys of animals, may it be human or dog. So this discovery was more or less accidental, as I've already explained, as he did not set out deliberately to make an organic synthesis to disprove vitalism. He was not trying to contradict Berzelius' theory of vitalism, and which was a notion championed by his own teacher, that is Johannes Jacob Berzelius, and he had no intention of doing so. But still, the believers of vital force theory did not believe this, because they said that the ammonium cyanate which was synthesized, in fact, isomerized to give us a structure which was urea. Later, Louis Pasteur during 1856-7 worked on fermentations and pointed out the importance of enzymes in this process. That is, fermentation is brought about in presence of enzymes. So if we are talking of fermentation of sugars, that it is the enzymes which will convert it into alcohol. The breakthrough in enzyme research was made in 1897 by Edward Buchner, who extracted enzyme zymase from yeast cells in crude form, which could ferment a sugar molecule into alcohol in vitro. So that means that in presence of enzyme zymase, Buchner was able to ferment, or was able to bring about fermentation of sugar, and he was able to convert it in alcohol in vitro, and this was not being carried out in a human body, or it was not happening in vivo. And then at the same time, Emil Fischer synthesized phenylhydrazine, a base that helped him to elucidate the structure of sugar molecules. So after seeing the work of pioneers who led to the synthesis of organic compounds and their relationship with biological process, I would be carrying the discussion further, and we will first familiarize ourselves with the four major molecules of life. And as I've already discussed in my previous lecture, the four major molecules of life are carbohydrates, lipids, proteins, and nucleic acid. Now, if on your screens you can see a typical structure of a cell, and we know that the components of cell are made up of these biological molecules. So let us see how are these components made and what are the functions of these components. So if we talk of the cell wall is made up of cellulose, which is a structural carbohydrate. And if we proceed to chloroplast, then we find that in chloroplast we have grains of starch. And what is starch? Starch is a storage carbohydrate. Now, if we go on to microtubules, so we find that these microtubules are proteinaceous in nature, and they are responsible of movement within the cells and that of cell. The next component is mitochondria, and mitochondria is made up of lipids, proteins, and DNA. And you know, mitochondria is also rightly called as the powerhouse of the cell, since ATP is synthesized here. The next important component of the cell are ribosomes, and we know ribosomes are made up of RNA and proteins, and we find that proteins are synthesized on ribosomes. Next we have a plasma membrane, and plasma membrane is made up of carbohydrates and lipids and proteins. The last very important component of the cell is the chromatin. Chromatin is found in chromosomes, and we find that chromatin has DNA and proteins. That is, chromatin is a part of chromosomes and it contains DNA and proteins. And we know that DNA is the genetic material of all living organisms. So now to continue the lecture further, I will now be starting with the discussion in very brief of those of biomolecules. And we will start with carbohydrates. We know carbohydrates are chemically defined as polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. So they may be simple carbohydrates like sugars, or they may be complex carbohydrates like cellulose, which is a structural carbohydrate, or it could be glycogen or starch, which are the storage carbohydrates. And if we talk of simple sugars, they are collectively called as saccharides. And saccharides comes from the Greek word saccharon, meaning sugar. And these saccharides have a free aldehyde or a ketone group, which acts as a reducing agent, and therefore these sugars are known as reducing sugars. Most of the monosaccharides and disaccharides are reducing in nature, the exception being sucrose. Sucrose is non-reducing because it does not have a free aldehydic or a ketone group. It does not have a potential aldehydic or a ketone group also. Now let us see the food sources which are rich in carbohydrates. As you can see on your screens, the cereals such as wheat and rice are rich in carbohydrates. Then there are fruits like papaya, apple, oranges are also rich in carbohydrates. Some vegetables like Brussels, kale, these are also rich in carbohydrates. So in our daily life, we can get if we are taking a balanced diet, we can get carbohydrates from these sources which are easily available. Now let us look into the biological function of carbohydrates. So carbohydrates, including sugars, are crucial to the biological processes of living organisms. Plants and algae produce millions of tons of carbohydrates each year through photosynthesis. And the main function of carbohydrates is to provide energy, particularly through glucose. The next class of biomolecules which are important are lipids. Lipids are organic substances that are insoluble in water but are soluble in organic solvents. Why are they insoluble in water? Because they are related to fatty acids. And we know that a fatty acid is made up of a tail which is composed of a non-polar hydrocarbon part, and it has a head which is polar in nature. So what we find that lipids are insoluble in water, and these lipids are utilized by the living cells. Now, what is the importance of lipids? Lipids play an important role in the cellular structure and are the chief source of energy. And you also know that they are the chief constituents of biological membranes in cells. That is, phospholipids are important constituents of membranes. The next class of compounds which are important are proteins. We say proteins are another class of indispensable biomolecules which make up around 50% of the cellular dry weight. Proteins are polymers of amino acids arranged in the form of polypeptide chains. And when we talk of the structure of proteins, we can classify it as 4 levels. That is primary structure, secondary structure, tertiary and quaternary. This can also be referred to as 1 degree, 2 degree, 3 degree and 4 degrees respectively. So as on your screens, now you can see the primary structure of protein. And what is primary structure of protein? In primary structure of protein, we find that the arrangement of amino acids is specific. That is, it involves the specific sequence of amino acids in a polypeptide chain. Say for example, as I have shown here, this is a dipeptide which is made up of say alanine and glycine. Now since we have 2 amino acids, we can have 2 arrangements. That is, in one arrangement, glycine can be arranged to alanine, and in the second arrangement, alanine can be arranged to glycine. And this change in the specific sequence of amino acids change the properties of the particular polypeptide or a particular protein. So the specific sequence of amino acids in polypeptide chain is very, very important. Now we go on to the secondary structure of a protein, which involves hydrogen bonds between sequence of amino acids in polypeptide chain folding in 2 ways. And because of this hydrogen bonding, polypeptide chain can be folded in 2 ways. One is the beta pleated sheets, and as you can see on your screens, and other is the alpha helix structure, which we shall be discussing in our subsequent lectures. Now we go on to the tertiary structure of a protein. So when we are talking of a tertiary structure of a protein, we are talking at the 3rd level, and this refers to the conformation of the molecule, or in other words, I can say it refers to a 3-dimensional shape of a protein with many more types of bonds between the alpha helix and the beta pleated sheets. That means the other types of bonds that we are talking of here would be the ionic bonds, dipole-dipole interactions, van der Waals forces, hydrogen bonds and disulfide ridges. But what you have to keep in mind that the tertiary structure of protein will only involve a single polypeptide chain. That means it will involve folding of only polypeptide chain, which can be beta pleated and or alpha helix, and there we will have different types of bonds which will stabilize the tertiary structure and give it its particular conformation. Now when we talk of the quaternary structure of the protein, that means we are talking of the 4th level. And when we are talking of the 4th level, in this case what we find that in quaternary structure is made up of more than 1 polypeptide chains, and these polypeptide chains are also called as subunits. For example, if we look into the structure of biomolecule, which is hemoglobin, then what we find that this hemoglobin is made up of 4 subunits, among which 2 are alpha, that is 2 are alpha and 2 are beta subunits. Now let us talk of the functions of proteins. So when we talk of functions of proteins, what we find that proteins play both structural and dynamic roles. That is, myosin is the protein that allows movement by contraction of muscles. That means it is present in our muscles. And the most enzymes are proteinaceous in nature. We know enzymes are the biocatalysts that bring about reactions in biological systems, and most of them are proteinaceous in nature. Now the last category of biomolecules that I am going to discuss are nucleic acids. And as you know, nucleic acids refer to the genetic material found in the cell that carries all the hereditary information from parents to progeny. And we know there are two types of nucleic acids, which are DNA, that is deoxyribonucleic acid, and RNA, which is ribonucleic acid. So what is the function of the nucleic acids? As I have already said, the main function of nucleic acids is the transfer of genetic information and synthesis of proteins by processes known as transcription and translation. In transcription, DNA passes the message to RNA, and during translation, RNA gives message in the form of proteins to synthesize proteins and enzymes. So both of these nucleic acids are very, very vital. One is involved in the genetic transfer of genetic material, and the other is involved in the, and if we talk of them together, they are involved in synthesis of proteins and enzymes. So both of them are involved in the transfer of genetic material. DNA is involved in transfer of genetic material, and RNA is involved in synthesis of proteins. The monomeric units of nucleic acids is known as nucleotide, and as I have already discussed, it is composed of a nitrogenous base, a pentose sugar, and phosphate. Nitrogenous bases are in the form of heterocyclic rings. As you can see the structure of a nucleotide here, what we find that nucleotide has a nitrogenous base. Here I have shown the structure of DNA. We have a nitrogenous base, which is adenine, and then we have a pentose sugar, which is then linked to phosphate. And the DNA and RNA differ in the pentose sugar. That is, in DNA we have deoxyribose sugar, whereas in RNA we have ribose sugar. So DNA and RNA form the genetic material of the cell, both of them. And how do they form the genetic material of the cell? Because the sequence of nucleotides in DNA forms the transcript or the periodic table of life. So these four molecules are the common basis of all the living organisms on this planet. So in brief, I have discussed with you the four important biomolecules, which are carbohydrates, proteins, lipids, and nucleic acids. These are very important for all the living organisms. In my next lecture, now I shall be discussing as to can we consider water as a biomolecule. And for this lecture, I had referred to books by Carey Francis, that is on organic chemistry, McMurray Joan, organic chemistry by Wade, and of course Principles of Biochemistry, that is Lehinger's Principles of Biochemistry, and you can also refer to these books for your further studies. Thank you. viewers. In my previous lecture on series, that is molecules of life, I had discussed in brief 4 molecules of life, that are carbohydrates, proteins, lipids and nucleic acids. Now I would like to discuss water as a biomolecule. But do you think it is justified to call water as a biomolecule? For this, let us study in depth the properties of water. And at the end of the lecture, let us conclude that is it right to call water as a biomolecule? We know that activity of all the molecules of life require a medium to act and interact. This medium must have all the required characteristics to allow the biochemical reactions without affecting the cellular environment. For this, water is the most important single compound which supports all living organisms on earth. How? We see that major structural component of cells and body fluids is water. Not only this, it is habitat to many plants, microbes and animals. It is the medium where the cellular, biomolecular, biochemical reactions can take place. Not only this, it maintains the molecular conformation and stable structure of biomolecules inside cytoplasm. Proteins and nucleic acids fold and attain their active 3D form in an aqueous medium. Transport medium in plants and animals is water. So in plants we see that xylem, that water transports water from xylem. It transports water from roots to various parts of the body, to various parts of the plant body. And from phloem, the food which is synthesized by leaves is transported to different parts of the plant. And in animals, it is the blood which transports oxygen to various parts of the body. So we find that water is important in transport medium, in transporting food, food and minerals from one part to the other part of a living organism, may it be plant or an animal. Now what we find that water ionizes to provide proton and hydroxyl ions to charge proteins, phospholipids and nucleic acids, and it also maintains specific pH ranges for proteins and enzymes to function. We know that enzymes are active at a specific pH, so it is water that maintains that specific pH. So we find that water is stabilizing with the help of H plus and OH ions. It is stabilizing proteins, phospholipids and nucleic acids, and it is also maintaining the pH for enzymes and proteins to function in a biological system. So that means it is acting as a solvent for various buffers in cell. Water, as you know, is also raw material in photosynthesis. We know in photosynthesis, carbon dioxide reacts with water in presence of chlorophyll in chloroplast to give us glucose, plus oxygen is liberated. So from where does this oxygen is liberated? It is the photolysis of water which evolves oxygen. And not only this, even when we are respiring, that when we use this oxygen to generate energy, what we find that this oxygen now breaks down glucose molecules during respiration, and there water is evolved, water is produced. And we've already discussed in the previous lecture that the hydrolysis reactions which are carried out in cells are brought about by water. That is, it is water which helps to break down, say, let us say, a peptide chain into simple amino acids. And it is the dehydration reactions, that is condensation reactions, which are involved in making polymers from different monomer units. For example, glucose molecules will react in such a way that the OH group of one molecule will react with the OH group of the second molecule of glucose, and in the process a glycosidic linkage is formed with elimination of water. So we see that water is involved everywhere in biological system. So now the question is, why is water so important? It is the properties of water that make it so important, and we can also consider it to be a biomolecule. And as you can see on your screens, the structure of water molecule, and what we find here, that in water oxygen is attached to two hydrogens. And now let us see what is the interaction between these molecules. We know. And now if we look at the molecular structure of water, what we find that electrons are, the distribution of electrons of a water molecule is uneven. That is, it is asymmetrical. That is, electrons are held more towards, they are distributed more towards oxygen. The reason being, oxygen is more electronegative than hydrogen, as a result of which oxygen will pull the electrons towards itself. So what we find, we can say that electrons spend more time near the oxygen and less time near hydrogen. So as a result of which, what happens is that electrons move towards oxygen, that is, they move away from hydrogen atoms. Therefore we say that the hydrogen end has a slightly positive charge and oxygen atom has a slight negative charge. As you can see on your screens here, that oxygen has attracted the electrons toward itself because of its high electronegativity, as a result of which oxygen has a slight negative charge and the positive charge is over the hydrogen atoms. So that means this molecule, now water, is polar in nature. Now since it is polar in nature, it interacts with other molecules which are necessary for life. And we know most of the substances in cell are floating around in water-based cytoplasmic environment. So what happens now, since the water molecule is polar in nature, water molecules can thus be oriented towards both positive and negative ions. Therefore, they behave as dipoles. And we say, since water is polar, why is water polar in nature? Because it has slightly charged regions. Whereas if we talk of a non-polar molecule like water, like hydrogen or oxygen, these molecules are non-polar because there is no distribution of charges. So what you can see in this slide, that oxygen has a slight negative charge on it and hydrogen has a slight positive charge on it, and it will now behave as a dipole. So the hydrogen bond, now when we talk of an hydrogen bond, so because it is behaving like a dipole, what we find that water is capable of forming hydrogen bond. Now before discussing how water forms hydrogen bond, let us discuss what is hydrogen bond. Hydrogen bond is unequal electron sharing, results in as an electrostatic interaction between a hydrogen atom attached covalently to an oxygen atom with the electrons of a second electronegative atom of oxygen to which it does not have any direct covalent bonding. So this is called hydrogen bonding. So as I've shown on your screens in simple language, what we find that this oxygen is attached to two hydrogens, and there we have another molecule of water. And what we find that this hydrogen makes hydrogen which is attached to an electronegative oxygen makes a bond with the second atom of oxygen, and this bond is known as hydrogen bond. So we can say hydrogen bond forms between slightly positive hydrogen atoms and slightly negative oxygen atoms. And what we find in this case that each water molecule makes hydrogen bonding with neighboring 4 molecules of water. So, and this hydrogen bonding here is known as intermolecular hydrogen bonding, that is, it is between 2 water molecules. And when we discuss the properties of water, it is that the hydrogen bond is very vital for the different properties of water. That is, we say that water has a special position. That is, so now let me say what is a hydrogen bond. So we can say a hydrogen bond is an attraction between 2 atoms that already participate in other chemical bonds. As you can see here, that here it is an interaction between 2 atoms, and these atoms are already involved in other linkages, in other bonds. That is, this oxygen is bonded to hydrogen here, and in hydrogen here in a water molecule, and now hydrogen is binding these 2 oxygens through a hydrogen bond. And when we talk of a hydrogen bond, then we know that the strength ranges from 4 kilojoules to 50 kilojoules per mole of a hydrogen bonds. And these hydrogen bonds were first described by Latimer and Rodebush in 1920. These hydrogen bonds, as I said, can be of varying strength. That is the strength may be 4 kilojoules to 50 kilojoules. So we can say that the strength of the hydrogen bonds is varying, it is different. So we can now have strong hydrogen bonds and weak hydrogen bonds. So when do we have a strong hydrogen bond in a molecule, and when do we have a weak hydrogen bond in molecule? When hydrogen bonds are strongest, when oxygen, hydrogen and oxygen are in a straight line. This is the ideal geometry for a strong hydrogen bond to be formed. But this ideal geometry is not possible in, say, proteins, where the hydrogen bonded moieties are constrained, that is, they may be bent. So as a result, bond is weaker. So as you can see, I have shown here a water molecule, and what we find that these here, that oxygen, this is here, the molecule is what? It is linear. So what we find that here, the oxygen, we say that what we find here is that oxygen, hydrogen and oxygen are in the straight line. So oxygen, what we have here, hydrogen and hydrogen here, they are in straight line, and since they are straight line, here the hydrogen bond will be strong. That is, the hydrogen bond between 2 water molecules will be strong. And similarly, we find that the hydrogen bond between, say, a water molecule, between an alcohol and a phosphate group, say in triglycerides, will also be strong, because their oxygen, hydrogen and oxygen are linear. But if this arrangement was not linear, it was bent, then in this case the hydrogen bond will be weak. So for a strong hydrogen bond to be formed, the hydrogen, oxygen and hydrogen must be linear, or we can say if the bond is between, say, NH2 and a OH group, that is between an amino group and water, then in that case we say that nitrogen, hydrogen and oxygen must be linear. So as I've said that hydrogen bonds play a very vital role in discussing the properties of water. So we say hydrogen is responsible for 4 hydrogen bonds are important for 4 important properties of water. That is, water can act as a universal solvent, then water has high specific heat, water shows surface tension, and then what we find that water, in spite of being liquid, is less dense as a that why is water less dense when it is in a solid state, whereas it should have been denser than liquid when water exists in liquid state. You know that water can exist in 3 forms: liquid, solid, that is solid, liquid and gas. And liquid state in case of water is more dense as compared to its solid state. So now let us look into the structures where we find hydrogen bonds in biological systems. So in this case what we find that normally when we talk of an hydrogen bond, we talk of an acceptor and a donor. So we can say when we are talking of a bond here, that is between a carbonyl group and a hydroxyl group, that is of a water molecule. In case of carbohydrates, so we can say in this case one molecule, that one atom of oxygen is a donor, and that is hydrogen is acceptor is usually. So we can say one of the atoms here is acting as an acceptor and one is acting as a donor. So here we have, I've shown a hydrogen bond as it may exist in heterocyclic bases, and then again here we have a hydrogen bond between a ether linkage and say a water molecule which may be present in carbohydrates. And then we again here we have shown hydrogen bonding say maybe between the oxygen of the carbonyl group and a amino group of say a protein, resulting in a form of which is found in peptide bonds. And then again we have a linkage between a oxygen and a nitrogen. Here there's a hydrogen bond, and then of course here's another hydrogen bond between an imide and a nitrogen. And both the cases here are nitrogen, which is an imide here, which may be present in a heterocyclic molecule, that is the bases which are present in nucleic acids, and we have a say amino acid here which could be adenine. So when we talk of hydrogen bonds, they can also, that means now hydrogen bonds can also form with a single large organic molecule. So once I like to go back here, so when we are talking of hydrogen bonds here, so what we find that hydrogen bonds, when I talk here, they can be, these hydrogen bonds here are between the complementary, they can be between the complementary bases. So now hydrogen bonds can also form a single large organic molecule. That means what I am trying to tell you here is that hydrogen bonds can be formed between different parts of a protein molecule and bend the molecule into a distinctive shape which is important for the function of proteins. Not only this, as I have discussed in my previous slides, that hydrogen bonds also hold together 2 nucleotide chains of DNA molecules, and what we find that here we have hydrogen bonds between complementary bases of DNA, that is between the thymine and the adenine, and these bonds I have already explained in my previous slides, that here we have a possibility of hydrogen bonding here, and these hydrogen bonds are formed between the complementary bases of DNA. Not only this, hydrogen bonds also explain why water's boiling point is 100 degrees, and this is higher than the boiling points of similar substances without hydrogen bonds. And if we look into the hydrides of this group, the boiling point of water should have been minus 100 degrees. But it is because of hydrogen bonding that the boiling point of water is 100 degrees Celsius. And because the boiling point of water is so high, most of the water exists in liquid state on earth, and you know that 75% of the earth is covered by water. And we know, now you can understand that why is water so important. This liquid water, water is needed by all living organisms. Therefore, the availability of liquid water enables life to survive over much of the planet. No wonder 75% of our planet is water. So now, what are the life providing properties of water? The unique properties of water are due to hydrogen bonds. So we say hydrogen bonds are central. Why are they central? Because hydrogen bonds in liquid water are central to water's life providing properties. That it is the hydrogen bonds which will keep water in liquid state, and we know most of the life will exist in water. So because of its extensive hydrogen bonding, water is liquid over a far greater range of temperatures that would be expected for a molecule of its size. If it was not for hydrogen bonding, water would not have existed as liquid. And we know that molecules of water are constantly moving in relation to each other, and the hydrogen bonds are continuously breaking and reforming. Imagine at a time scale faster than 200 femtoseconds, that is 2 to the power of 10 minus 13 seconds. So that means hydrogen bonds, when they are forming so quickly, that is, a hydrogen bond is formed in 200 femtoseconds, it is formed and it is broken. Therefore, we do not understand the importance. That means we do not comprehend that there is a hydrogen bond, and it is because of, and therefore, and we say that these hydrogen bonds are strong enough, although they are breaking and forming so quickly, these hydrogen bonds are strong enough to create many of the peculiar properties of water, some of which make it integral to life. So when we are talking of a hydrogen bond, we see that a single hydrogen bond is weak, but the sheer number makes it a strong force. So now we can use water as a universal solvent. Why does water act as a universal solvent? We know water is a polar molecule and has exceptionally high hydrogen bonding ability. This unique property allows it to dissolve a variety of molecules like sugars, amino acids, carrier proteins in solution. And what we find that, as I've already said, I've already shown that there is hydrogen bond is between the hydroxyl group of we can say of a carbohydrate and a water molecule. Then it can be between the carbonyl group of a carbohydrate or a amide and a water molecule, and as I have shown here, and that is here it is between the amido group and the between here we have hydrogen bond between the two peptide chains also. They are bonded. So now we see that the sugars and proteins which are containing OH or NH2 groups therefore dissolve easily in water, as you can see here that sugars can form hydrogen bonding here with water. Here the carbonyl group of a say a sugar molecule is forming a hydrogen bond. Then what we find that proteins also can form hydrogen bonds because proteins have amino groups, And because of these polar groups, that is OH in sugars and amino group in proteins, these molecules are equally soluble in water, easily soluble in water, they dissolve in water. Therefore, we say that an aqueous medium would be highly suitable for uptake of dissolved mineral nutrients. So we say that so it is very vital, water is very vital for growth and development of plants, and it is also very important for animals. So what happens now? How does it act as a solvent? What we find that if we talk of a normal ionic compound, say NaCl, so what we find that ionic compounds disperse as ions in water and get evenly distributed. An aqueous solution is formed when the one substance dissolves in another. That means when say I can say NaCl dissolves in water, so we call it as an aqueous solution, and this solution is a homogeneous mixture, as you can see here. That is solute here we have taken as water, solvent as water, and solute say as NaCl molecule, which will be soluble in this water. How does this happen? So what happens is water dissolves many crystal salts by hydrating their compound ions. So what happens is when we are talking of NaCl, water clusters around the ions Na, sodium and chloride, and the crystal lattice of NaCl is disturbed. And now what we find that the ionic charges are partially neutralized on these ions, and the electrostatic attractions between them are weakened, and the lattice structure is destroyed. This is what you can see on your screens, that when we have a molecule, a crystal of NaCl, the water molecule will surround the Na molecules, Na ions, sodium ions, by its oxygen and the chloride ions will be stabilized through hydrogen bonding. They are hydrated as chlorides. So what we find that because of water, the electrostatic forces between Na and Cl are weakened, and this crystal lattice breaks. And what happens when this crystal lattice breaks? There is greater freedom of movement, and when there is greater freedom of movement, that means the randomness is increasing, or in other words, the entropy of the system is increasing, and more NaCl gets dissolved in water. Now the last importance, the last example where I would like to discuss the importance of water is the enzyme substrate interaction. So when does an enzyme substrate interaction occur? What happens is when an enzyme substrate complex is formed, it is due to the increase in entropy. We know that when both the enzymes and the substrate are existing separately, they will force the neighboring water molecules into an ordered shell. So what we find here, they are in ordered shell. Now as you can see on your screens, that when this substrate comes, when the enzyme comes near to the substrate, the sum of the ordered water gets released. You can see that the water here is getting released, and when the water is getting released here, the entropy or the randomness of the system increases, and this gives a thermodynamic push for the formation of enzyme substrate complex. So what we find that water is playing a very pivotal role. It is playing pivotal role as to in the transport of minerals, transport of blood, in the transport system in animals and plants. It is acting as a solvent. It is helping to bring about the reaction between a substrate and an enzyme. It is stabilizing the DNA, RNA, carbohydrate molecules. It is also important in lipids. So we can say that we can rightly call water as a biomolecule. So along with carbohydrates, proteins, lipids, nucleic acids, we can also call water, which is an inorganic molecule, as a biomolecule. As you've already seen, it plays a pivotal role and it is essential for existence of life on this planet. Thank you.
In this lesson, we will learn about nature of enzyme. Introduction. The living cell is the site of tremendous biochemical activity called metabolism. This is the process of chemical and physical change which goes on continually in the living organism. Build-up of new tissue, replacement of old tissue, conversion of food to energy, disposal of waste materials, reproduction, all the activities that we characterized as life. Building up and tearing down takes place in the face of an apparent paradox. The greatest majority of these biochemical reactions do not take place spontaneously. The phenomenon of catalysis makes possible biochemical reactions necessary for all life processes. Catalysis is defined as the acceleration of a chemical reaction by some substance which itself undergoes no permanent chemical change. The catalysts of biochemical reactions are enzymes and are responsible for bringing about almost all of the chemical reactions in the living organisms. Without these enzymes, the reactions take place at the rate far too slow for the pace of metabolism. The oxidation of fatty acid to carbon dioxide and water is not a gentle process in a test tube. Extremes of pH, high temperatures and corrosive chemicals are required. Yet in the body, such a reaction takes place smoothly and rapidly within a narrow range of pH and temperature. In the laboratory, the average protein must be boiled for about 24 hours in a 20% HCl solution to achieve a complete breakdown. In the body, the breakdown takes place in 4 hours or less under conditions of mild physiological temperature and pH. It is through attempts at understanding more about enzyme catalysts, what they are, what they do, and how they do it. That many advances in medicine and the life sciences have been brought about. We will have a glance over some early enzyme discoveries. The existence of enzymes has been known well for over a century. Some of the earliest studies were performed in 1835 by the Swedish chemist John Jacob Berzelius, who termed their chemical action as catalytic. The first enzyme was obtained in pure form in 1926 by James B. Sumner of Cornell University. Sumner was able to isolate and crystallize the enzyme urease from the jack bean. For his work, he earned the 1947 Nobel Prize. In sharing with John H. Northrop and Wendell M. Stanley of the Rockefeller Institute of Medical Research, Northrop and Stanley discovered a complex procedure for isolating pepsin. This precipitation technique has been used to crystallize several enzymes. Let's explore the chemical nature of enzymes. All known enzymes are proteins. They are high molecular weight compounds made up of chains of amino acids linked together by peptide bonds. Enzymes are proteins made up of amino acids. Active sites are formed by secondary and tertiary clustering of amino acids, which results in the formation of physical grooves where the substrate binds. Charges on amino acids allow them to interact with substrate by donating or accepting electrons or protons depending on the enzyme. Enzyme substrate binding involves temporary H positive and or ionic bond at the active site. Enzymes can be denatured and precipitated with salts, solvents and other reagents. They have high molecular weights ranging from 10,000 to 20 lakhs. Many enzymes require the presence of other compounds, that is, cofactors. Before their catalytic activity can be exerted, this entire active complex is referred to as holoenzyme. That is, apoenzyme means the protein portion and the cofactor means the coenzyme, prosthetic group or metal ion activator. According to Holm, the cofactor may be, first one is coenzyme. It is a non-proteinaceous organic substance which is thermostable and loosely attached to the protein part. Second is prosthetic group. It is an organic substance which is thermostable and firmly attached to the protein or apoenzyme portion. Third one is metal ion activators. It includes ions like K positive, Fe2 positive, Fe3 positive, Cu2 positive, CO2 positive, Zn2 positive, Mn2 positive, Mg2 positive, Ca2 positive and MO3 positive. Now we will study naming and classification of enzymes. Enzyme Commission has developed a rule for naming enzymes. According to this rule, each enzyme is classified and named according to the type of chemical reaction it catalyzes. The Enzyme Commission has given each enzyme a number with 4 parts like EC 2.7.1.2, that is hexokinase. The first 3 numbers define major class, subclass and sub-subclass respectively. The last number in a serial number is the sub-subclass, indicating the order in which each enzyme is added to the list. Common name and EC numbers of some enzymes are alcohol dehydrogenase, EC number 1.1.1.1, phosphofructokinase, EC number 2.7.1.11, glutamine synthetase EC number 6.3.1.2, acetylcholinesterase EC number 3.1.1.7, except for some of the originally studied enzymes such as pepsin, renin and trypsin, most enzymes names end in ASE. The International Union of Biochemistry initiated standards of enzyme nomenclature which recommended that the enzyme names indicate both the substrate acted upon and the type of reaction catalyzed. Under this system, the enzyme uricase is called urate O2 oxidoreductase, while the enzyme glutamic oxaloacetic transaminase is called L-aspartate 2-oxaloglutarate aminotransferase. Enzymes can be classified by the kind of chemical reaction they catalyzed. First one is addition or removal of water, hydrolysis. These include esterases, carbohydrases, nucleases, deaminases and proteases. Hydrases such as fumarase, enolase, acotinase and carbonic anhydrase. Second is transfer of electrons from oxidases, dehydrogenase. Third one is transfer of radical transglycosidases of monosaccharides, transphosphorylases and phosphomutases of phosphate group, transaminases of amino group, transmethylases of methyl group, transacetylases of acetyl group. Fourth is splitting or forming a carbon-carbon bond, desmolases. Fifth one is changing geometry or structure of molecule, isomerases. Last one is joining two molecules through hydrolysis of pyrophosphate bond in ATP or other triphosphate, ligases. Moving ahead to enzyme kinetics. Here we will firstly study basic enzyme reactions. Enzymes are catalysts and increase the speed of chemical reaction without themselves undergoing any permanent chemical change. They are neither used up in the reaction nor do they appear as reaction products. The basic enzymatic reaction can be represented as substrate plus enzyme gives rise to product plus enzyme. A theory to explain the catalytic action of enzymes were proposed by the Swedish chemist Svante Arrhenius in 1888, proposed that the substrate and enzyme formed some intermediate substance which is known as the enzyme substrate complex. The reaction can be represented as substrate plus enzyme gives rise to enzyme substrate complex. If this reaction is combined with the original equation 1, then the reaction becomes substrate plus enzyme gives rise to enzyme substrate complex, which again gives rise to product plus enzyme. Two important models have been proposed to describe the binding process of enzyme to substrate. The first, lock and key model, proposed by Emil Fischer in 1890. According to this model, the active site of unbound enzyme is complementary in shape to that of substrate. That is, the specificity of binding depends on the precisely defined arrangement of atoms in an active site. To fit into the site, a substrate must have a matching shape. However, it is now evident that the shape of the active sites of many enzymes are markedly modified by the binding of substrate, as was postulated by Daniel E. Koshland Jr. in 1958. The active sites of these enzymes assume shapes that are complementary to that of the substrate only after the substrate is bound. The process of dynamic recognition is called induced fit model. Let's see how it works. Enzyme-substrate interaction involves multiple bonds, individually weak, readily reversible. Upon substrate binding, enzyme changes conformation at active site to promote catalytic function. Change in conformation ensures specificity to fit, also allows for the activation of substrate. Three mechanisms by which this occurs are bond distortion, enzyme donates or accepts protons, enzyme donates or accepts electrons. Temporary bonds between substrate and enzyme form transient enzyme-substrate complex. Intermediate step in catalytic event, product then released, enzyme reverts to initial conformation. Now let's discuss specificity of enzymes. One of the properties of enzymes that makes them so important as diagnostic and research tools is the specificity that exhibit relative to the reaction they catalyze. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are four distinct types of specificity. The first one is absolute specificity. The enzyme will catalyze only one reaction. Second is group specificity. The enzyme will act only on molecules that have specific functional groups such as amino, phosphate and methyl groups. Third is linkage specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. And the last is stereochemical specificity. The enzyme will act on a particular steric or optical isomer. Though enzymes exhibit great degrees of specificity, cofactors may serve many apoenzymes. For example, nicotinamide adenine dinucleotide is a coenzyme for a great number of dehydrogenase reactions in which it acts as a hydrogen acceptor. Among them are the alcohol dehydrogenase, malate dehydrogenase and lactic dehydrogenase reactions. Knowing about energy levels, chemists have known for almost a century that for most chemical reactions to proceed, some form of energy is needed. They have termed this quantity of energy the energy of activation. It is the magnitude of activation energy which determines how fast the reaction will proceed. It is believed that enzymes lower the activation energy for the reaction they are catalyzing. The enzyme is thought to reduce the path of the reaction. This shortened path would require less energy for each molecule of substrate converted to product. Given a total amount of available energy, more molecules of substrate would be converted when the enzyme is present, that is the shortened path, than when it is absent. Hence, the reaction is said to go faster in a given period of time. Elaborating factors that affect enzyme activity. Several factors affect the rate at which enzymatic reaction proceed, like temperature, pH, enzyme concentration, substrate concentration and the presence of any inhibitors or activators. First one is effect of temperature. Since most of the enzymes are proteins, so they unfold and denature at high temperature. This denaturation causes a decrease or loss of catalytic activity, which in turn decreases the rate of reaction. Similarly, low temperature also inactivates the enzyme. Within limits, the rate of an enzyme catalyzed reaction increases with increase in temperature because the greater kinetic energy of both enzyme and substrate molecule ensures more frequent collisions. Thus, an enzyme catalyzed reaction achieves its maximum reaction at an intermediate temperature called optimum temperature. The optimum temperature varies greatly from enzyme to enzyme and especially from organism to organism. The reaction rate of most enzymes of homotherms are maximum at about 40 degrees centigrade. Some enzymes retain activity at unusual high temperature. For example, enzymes like Taq DNA polymerase present in thermophilic bacteria Thermus aquaticus has optimum temperature 72 degrees centigrade. Second is effect of pH. Enzymes are sensitive to pH. This pH dependence is due to the presence of charged amino acids at the active site. Variation in pH cause changes in overall charge of amino acids. Thus, enzymes have its optimum pH at which rapid activity occurs. For example, optimal pH for pepsin, that is protein digesting enzyme, is 2 and for trypsin it is 8.5. Optimum activity is generally for most of the enzymes are observed between pH values 5 to 9. When the activity is plotted against pH, a bell-shaped curve is usually obtained. Third is enzyme concentration. In order to study the effect of increasing the enzyme concentration upon the reaction rate, the substrate must be present in an excess amount, that is, the reaction must be independent of the substrate concentration. Any change in the amount of product formed over a specific period of time will be dependent upon the level of enzyme present. Fourth is substrate concentration. If we examine the effect of varying the substrate concentration on the rate of enzyme catalyzed reaction, when enzyme concentration is kept constant, it is found that at relatively low concentration of substrate, the rate of reaction increases in proportion to the concentration, but as the concentration is increased further, the reaction rate is increased by smaller and smaller amounts. Finally, when the substrate concentration is raised still higher, a point will be reached beyond which there is no further increase in the reaction rate. At this stage, even if the high substrate concentration is added, the reaction rate will remain essentially constant. At this stage of reaction, it is called the maximum velocity or maximum rate, which is designated as Vmax. All enzymes exhibit the saturation effects. A general theory of enzymes was proposed by Michaelis and Mentene, who also proposed the mechanism of formation of enzyme substrate complex on this basis. From the saturation curve, an equation was derived which is known as Michaelis constant, which specifies the quantitative relationship between substrate concentration and Vmax for different enzymes. It is defined as the substrate concentration at which a given enzyme yields 1/2 its maximum velocity, V by 2 equals to Km. Value of Km is important in tending out the affinity of enzymes with that of substrate. Lower the Km values, higher the affinity. Higher the Km values, lower the affinity. This relates relationship between substrate and enzymes. The Km values of different enzymes differs depending upon the condition at which enzymes reaction takes place. It is one of the important characteristic of enzymes. The characteristic shape of the substrate saturation curve for enzymes can be expressed mathematically by Michaelis-Mentene equation. Here, Vmax represents the maximum rate achieved by the system at maximum substrate concentration. The Michaelis constant Km is substrate concentration at which the reaction rate is half of Vmax. Biochemical reactions involving a single substrate are often assumed to follow Michaelis maintained kinetics without regard to the model's underlying assumptions. Equation describing relationship between rate of reaction and substrate concentration for enzyme catalyzed reaction. And lastly, we discuss enzyme inhibition. Inhibition of enzyme activity may be irreversible or reversible. First one is irreversible inhibitors. Irreversible inhibitors usually bound covalently to the enzyme and destroy the functional group and active site. Most of irreversible inhibitors are toxic substances. The action of nerve gases on acetylcholinesterase, an example that plays an important role in the transmission of nerve impulses, is an example of irreversible inhibition. Second is reversible inhibitors. In reversible inhibition, the inhibitor can dissociate from the enzyme. Reversible inhibitors involve the non-covalent binding with enzymes. Two common types of reversible inhibitors are: the first one is competitive inhibition. The structure of competitive inhibitors closely resembles that of the enzyme's normal structure. Example: in case of methanol poisoning, methanol in the body is converted to harmful formaldehyde by alcohol dehydrogenase. A high dose of ethanol is used to alleviate the effect of methanol because ethanol competitively binds with the active site of alcohol dehydrogenase. Second is non-competitive inhibition. In non-competitive inhibition, the inhibitors binds to the enzyme at a site other than the active site. Inhibitors binding alter the enzyme's three-dimensional configuration and blocks the reaction. Metal ions including copper, mercury and silver act as a non-competitive inhibitors of many enzymes by binding reversibly to the SH group of cysteine, the amino acid that forms disulfide bridges. This disrupts the structure of enzyme.
Thus, at the end, to summarize, the chapter started with introduction to early enzyme discoveries, followed by understanding of chemical nature of enzymes. Naming and classification of enzymes according to the rules of enzyme commission and the type of chemical reaction they catalyzes has also been studied and cited. Enzyme kinetics with its reaction has been elaborated. Specificity of enzymes, a very important diagnostic and research tool, is also discussed. Energy levels have been described specifically as they are very important for most of the reaction to proceed. Several factors that affect enzyme activity like temperature, pH, enzyme concentration, substrate concentration and the presence of any inhibitors or activators have also been mentioned. Lastly, we have seen enzyme inhibition with its types and examples.
microbes that grow in or on the culture medium are known as microbial cultures. Most of the microbes are able to grow on this synthetic medium, but however, there are certain group of microbes which fail to grow in lab conditions. Now let us move to the types of culture medium. The type or the classification of culture medium can be done in various ways. The first system or the type of classification is based on its consistency. What is consistency? That is the physical appearance of culture medium. The second way in which we can classify the culture medium is on the basis of its nutritional components. Means what kind of nutrients have been added to the medium. The third system or type of classification of culture medium is based on the functional use. Let us come to the first system of classification of culture medium, that is consistency, which means the physical appearance of the culture medium. If the culture medium is liquid in nature, then it is known as broth. The liquid culture is done when we require a large number of population of bacteria in a small defined period of time. Then we go for liquid culture. The solid medium is done by addition of agar. Agar generally is added at a concentration of 1.5 to 2% to the culture medium. It is then sterilized by means of autoclave. If the solid medium is dispensed into culture tubes and hardened in a slanted position, then it is known as agar slants. The slants are generally used for maintenance of pure culture and for biochemical studies of that particular microbe.
Welcome to the series of lectures on textile fibers. So far we had been discussing the textile fibers, both natural and man-made. Now we come to the next step, that is formation of yarn from textile fibers. Now, what is a yarn? A yarn can be defined as a continuous strand of textile fibers which can be both staple or filament in a form of strand which is suitable for weaving, knitting or otherwise intertwining to form a textile material. Now a yarn can be constructed as an assemblage of textile fibers and which acts as a unit in fabric formation. Now what are the characteristics of yarns? To be a yarn, it should have certain specific characteristics. It should have certain strands of fibers which need to be twisted together to form a continuous single strand. Now each strand in turn is made of small fibers. These short fibers are spun into longer yarns or filaments. Now long continuous strands may only require additional twisting because as it is, they are longer filaments, longer strands, slight twisting may form a yarn. Now yarns can be classified into simple yarns, novelty yarns and cabled yarns. Simple yarns are made up of natural or man-made fibers with same length and with same properties. Simple yarns can further be divided into first single ply yarns which are made from a group of filaments or staple fibers twisted together. A slightly higher twist is given to a single ply yarn. Multiply yarn is made up of twisted or piled two or more single yarns together. They can be given S twist or Z twist. We shall be further coming to S type of twist and Z type of twist. Now cord yarns are made by twisting together two or more ply yarns. Now on your screen you can see the S type of twist that is given. Now these four strands are joined together, they are twisted by giving a S
type of twist. Now each ply is called a single because it can't be broken down into more strands. If you try pulling out one of the singles apart, you get loose fibers. So each of these strands that is visible on your screen is made up of loose fibers. Now 4 strands, they join together, they are twisted together, given a S type of twist to form this yarn. The final yarn is twisted in the S direction. Now this means if you overlay the letter S on it, the center part of letter S or the middle bar of the letter mimics the direction of the final twist. The arrow, this arrow on the screen that you can see, also points in this direction. Now this is the Z twist. Conventionally, S-plied yarns are made into thicker yarns by doing one or more twisting in the Z direction. This is how a cabled yarn is made. A cable, as you all know, is much more thicker than a normal yarn. A number of plies or plied yarns are twisted together, given this Z type of twist, to form a cable. The final yarn has the look of a cabled cord. All this twisting results in firmly spun yarn. The final yarn, which has its twist in the Z direction, consists of 4 strands. In the diagram here, you can see 1, 2, 3 and 4 strands. And individually, each strand is also further made of 2 plies. So it means each strand of 2 plies and then 4 strands of 2 plies each, forming this cable with a Z twist. The center part of the Z, as the arrow shows, is the direction of the twist. Now what is the effect of twist on staple yarn properties? Why do we twist the yarn? So as twist increases, the yarn strength and yarn stiffness also increases up to a certain point. But if you keep twisting the yarn further, the strength may later on decrease. Yarns with extremely high level of twist are called crepe yarns. As we see the crepe fabric, it is slightly crinkly. It gives a crinkled effect because of the high twist that is given to the yarn. Now, these yarns, they possess a high degree of torque, which causes them to buckle, and the decrease in length is there. The total fabric, after it is made and given a higher twist and crepe fabric is formed, the overall length of the fabric decreases. Now woven fabric containing these yarns have a pebbled surface texture. It means the surface of the fabric is not very smooth. It is not very lustrous either, but it has a pebbly surface because of the higher twist that is given to the yarn. Now second type of yarns are the novelty yarns. The novelty yarns are the decorative yarns which are made thus to look more attractive and beautiful. Sometimes more decorative elements are added to the spinning process. There may be a third yarn or a second yarn which can be added during the spinning process to give luster or additional look or additional color to the yarn. Third type of yarns are the texture yarns. These yarns are made up of either filament yarns or staple yarns. Textured yarns offer a variety of appearances, and mostly textured yarns, they give bulk to the fabric or fluff to the fabric, and sometimes they can be smooth or fine also. Now we come to the history of yarns. How did yarn making originate, and what are the processes it went through over a period of time? Firstly, natural fibers, cotton, flax, silk and wool represent the major fibers available to the ancient civilizations. Because man-made fibers, they came much later. Initially, man started using the natural fibers. The earliest known samples of yarn and fabric of any kind were found near Robenhausen in Switzerland, where bundles of flax fibers and yarns and fragments of plain weave linen fabric were estimated to be about 7,000 years old. Now cotton has been cultivated and used to make fabrics for at least 7,000 years. It may have existed in Egypt as early as 12,000 BC. Fragments of cotton fabrics have been found by archaeologists in Mexico from 3500 BC, in India 3000 BC, in Peru 2500 BC, and in the southwestern United States 500 BC. These are certain historical perspectives which show when and where the fabrics were seen by the archaeologists. Silk culture remained a speciality of Chinese from its beginning, that is 2600 BC, until the 6th century, when silkworms were first raised in the Byzantine Empire. The history says the silkworms, they were smuggled out of China, and later on the silk culture developed all over the world. Now, synthetic fibers did not appear until much later. The first synthetic fiber, rayon, made from cotton or wood fibers, was developed in 1891, but not commercially produced until 1911. It means the synthetic fibers did not exist before 20th century. Until about 1300 years, yarn was spun on the spindle and wall. A spindle is a rounded stick with tapered ends to which the fibers are attached and twisted. A wall is a weight attached to the spindle that acts as a flywheel to keep the spindle rotating. This was the ancient process by which the yarn was produced. A major improvement was the spinning wheel invented in India between 500 and 1000 AD and first used in Europe during Middle Ages. About 150 years later, the Saxon wheel was introduced. Operated by a foot pedal, the Saxon wheel allowed both hands the freedom of work with the fibers. Now, a number of developments during the 18th century further mechanized the spinning process. In 1733, the flying shuttle was invented by John Kay, followed by Hargreaves' spinning jenny in 1766. Several years later, Richard Arkwright patented the spinning frame, a machine that used a series of rotating rollers to draw out the fibers. A decade later, Samuel Crompton's mule machine was invented, which could spin any type of yarn in one continuous operation. The ring frame was invented in 1828 by the American John Thorpe and is still widely used today. This system involves hundreds of spindles mounted vertically inside a metal ring. Many natural fibers are now spun by the open-end system, where the fibers are drawn by air into a rapidly rotating cup and pulled out on the outer side or the other side as a finished yarn. After the historical perspective, we meet again after the break to discuss the formation of yarn from the fibers. We'll be talking about various steps, blending, carding, combing, and the different steps which are involved in the formation of yarns. Now we shall be talking about the yarn manufacturing process, that is the preparation of the fibers. Fibers are shipped from all over the country or various countries and are opened by hand or machine. Now, natural fibers, they do require certain cleaning, whereas synthetic fibers, they only require separating. Blending of different staple fibers may be required for certain applications. Now, blending may be done during formation of the lap, during carding, or during drawing out. Quantities of each fiber are measured carefully and their proportions are consistently maintained. Now blending is done also to mix two types of fibers. If we need the properties of two fibers, for example, cotton and polyester, if we are manufacturing polycot yarns, so blending of cotton and polyester will be done in fixed amounts, 40% cotton or 60% polyester, or it can also be 50% cotton or 50% polyester, and they need to be blended together thoroughly to form a blended yarn. Now this is the blending feeder. On this you can see the different types of fibers. They are put together. These fibers, they pass through these rollers, they are thoroughly mixed, and this is where we obtain the mixed or the blended fibers. Now the second step on your screen you can see is the removal of trash. Trash is the dirt particles or dust or even leaf particles which are present in the fibers, and more so in natural fibers. Cotton has considerable amount of trash in it. So this is the picker where all the fibers are laid, and they pass through these rollers up and down, and these screen rollers, as the fibers passes through them, the trash is seen falling down. You can see this is the trash which is fallen down below and is collected, and these are fans here which blow into these rollers, and when the fiber reaches the final stage, it takes the form of a lap and it is a cleaner fiber. Now next step is the carding process. The carding machine is set with hundreds of fine wires that separate the fibers and pull them into somewhat parallel form. Carding means the fibers, they are laid parallelly to each other. They are not in a haphazard form. A thin web of fiber is formed, and as it moves along, it passes through a funnel-shaped device that produces a rope-like strand of parallel fibers. It means a thick rope of fibers is formed at this stage. Blending can take place by joining laps of different fibers. A lap of one fiber can be formed, say cotton. A lap of second fiber, say wool, can be formed, and these two laps can be mixed together at this stage also. This is the carding machine in front of you. This is the first step where the fibers are fed into the rollers. They pass through various rollers over this large cylinder, and this is the Doffer comb. A comb is like a normal comb which we use for our hair. This comb combs the fibers and lays them parallel to each other, and then later they take the form of a rope. This rope is collected in this can here. After the carding process comes the combing process. When a smoother, finer yarn is required, fibers are subjected to further parallel method. Now, for certain types of fabrics, we do not require the combing process. But when we need the finer fabric, say for example in wool, when we are manufacturing worsteds, we go through this combing process. A comb-like device arranges the fibers into further parallel form with short fibers falling out of the strand. This is the place where shorter fibers, they are separated from comparatively longer fibers. After carding and combing process, the slivers are formed. Several slivers are taken together from different cans. You can see 6 slivers here from 6 different cans. They are taken together. They pass through these spoons, and then together they pass through these rollers. From these rollers they rotate at different speeds in different directions, and they are 6 times faster as the initial set. It means that these last rollers, they move at a faster speed than the rollers which are here initially. And these 6 strands, they are given a twist and they elongate, and in the form of a roving they come into this can. Now what is the drawing out process? After carding or combing, the fiber mass is referred to as the sliver. A single rope is called a sliver. Several slivers are combined before this process. A series of rollers rotating at different rates of speed elongate the sliver into a single more uniform strand that is given a small amount of twist and fed into larger cans. As I explained earlier, the slivers are much thicker. After the drawing out process, they become much finer and thinner. And also, another thing that is done here is they are given a slight twist and then they are fed into different cans. Carded slivers are drawn twice after carding. Combed slivers are drawn once before combing and twice more after combing. Twisting. The sliver is fed through a machine called a roving frame, where the strands of fiber are further elongated and given additional twist. These strands are now termed as roving. Earlier they were slivers. Now after the twist is given, we call them roving. Now these are the roving frames. From the can you can see the sliver coming out, and slivers, they pass through sets of rollers which rotate at a faster speed, and it comes to the roving frame, which also further gives the twist to the fiber. And on this you can see on the roving frame is the roving that is visible. Now this is the complete yarn manufacturing process. Now here the baling is done, the bales are opened, mixing of fibers is done, and the clean fiber is collected here in this can. And later on, this fiber is taken in the form of slivers, and over the revolving cylinders, these slivers, they pass through wire brushes, and these slivers are passed over these rollers, and they are carded, and then they are drawn out to form the final yarn. Now the steps of the yarn manufacturing process. We will revise the whole thing stepwise. First, the fibers are shipped in bales which are opened by hands or machines. Secondly, the picker loosens and separates the lumps of fibers and also cleans the fibers. Third, the carding machine separates the fibers and pulls them into a parallel form. The thin web of fibers formed then passes through a funnel-shaped device that produces a rope-like strand of parallel fibers. Rollers elongate the strand, called a sliver, into a single, more uniform strand that is given a small amount of twist and fed into larger cans. Now the final stage, the predominant commercial system of yarn formation are ring spinning and open-end spinning. In ring spinning, the roving is fed from the spool through rollers. These rollers elongate this roving further, which passes through eyelets moving down. The sliver is fed through a machine called a roving frame, where the strands of fiber are further elongated and given additional twist. It means the fibers, the roving becomes much more finer, it becomes much more thinner, and further twisting is done at this stage. Open end spinning omits the roving step. Now this is the spinning machine. The fibers, they are coming from here. This is the roving that is coming, and it passes through these rollers, and further this is the eyelet through which this roving is passing, and it comes on to the spinning frame, where a high degree of twist is given to the yarn and much finer yarn is obtained. Now, in the spinning process itself, different types of yarns can be formed. We can have a single ply yarn, we can have a double ply yarn, or even a cabled yarn can be formed. In this diagram here, you can see the fibers twisted into a single yarn. This is a single yarn, and these fibers are twisted here. And different thickness of twist can be taken. 2 single yarns twisted into ply yarns, and then this 1, 2 and 3 ply yarns, they are twisted together to form a cabled or a corded yarn. So this is how a corded yarn is made. During the spinning process, after the ply yarns are made, they can be twisted together to form a much thicker yarn depending upon the requirement of the yarn thickness. Now what is the future of spinning process? Spinning systems and yarn manufacturing machinery has become very advanced and very developed and more automated, and is integrated as a part of manufacturing unit rather than as a separate process. Spinning machines have already been developed that combine carding and drawing processes together. Production rates have increased in magnitude as machines become available with more spindles. A number of spindles, they function at the same time. And now robotic machines are also available, and the equipment is controlled by computers. Another continuing challenge for the industry is compliance with stricter environmental regulations. Recycling is also an issue, and processes are under development to manufacture yarn from scrap material, including denim. A lot of waste material is there during the spinning process which cannot be utilized for yarn formation. This leftover fiber and the material that is there can be recycled and reused for formation of different types of materials. Yarn producers will have to incorporate pollution prevention measures to meet the air and water quality restrictions. Textile industry as it is known for pollution creation. Therefore, the norms are much more strict for textile industry, and the textile industry will have to adhere to the prevention norms and ecological considerations. Genetic engineering will become more widely used for developing fibers with unique properties. Researchers have developed genetically altered cotton plants whose fibers are specially good at retaining warmth. Researchers are also working at developing colored cotton. The plants, they will be producing colored cotton fibers which will not require further processing, therefore reducing the overall process of textiles. This was the formation of yarns from natural fibers. In forthcoming lecture, we will be talking about the formation of yarns from synthetic fibers.
Microbes that grow in or on the culture medium are known as microbial cultures. Most of the microbes are able to grow on the synthetic medium, but however, there are certain group of microbes which fail to grow in lab conditions. Now let us move to the types of culture medium. The type or the classification of culture medium can be done in various ways. The first system or the type of classification is based on its consistency. What is consistency? That is the physical appearance of culture medium. The second way in which we can classify the culture medium is on the basis of its nutritional components. Means what kind of nutrients have been added to the medium. The third system or type of classification of culture medium is based on the functional use. Let us come to the first system of classification of culture medium, that is consistency, which means the physical appearance of the culture medium. If the culture medium is liquid in nature, then it is known as broth. The liquid culture is done when we require a large number of population of bacteria in a small defined period of time. Then we go for liquid culture. The solid medium is done by addition of agar. Agar generally is added at a concentration of 1.5 to 2% to the culture medium. It is then sterilized by means of autoclave. If the solid medium is dispensed into culture tubes and hardened in a slanted position, then it is known as agar slants. The slants are generally used for maintenance of pure culture and for biochemical studies of that particular microbe.
I'll particularly sing this in notation form.
Fine.
So that we can directly relate to the audience.
Yes.
Hello friends, welcome to a lecture on use and overutilization of surface and groundwater. To begin with, let us try and understand the importance of water. Water is a crucial natural resource. Its availability greatly influences the health of the people and development potential of the area. The National Water Policy 1987 states that water is a prime natural resource, basic human need, and precious national asset. It gives special attention to drinking water for both humans and animals over its other uses. And the policy calls for control on the exploitation of groundwater through regulation and an integrated and coordinated development of surface and groundwater. So far as we know that people all over the planet are dependent on water to grow food, generate power, cool the machines of industry, and carry wastes and much more. People also use water in their personal lives for bathing and cleaning, recreation, drinking, cooking, gardening, and just for the pleasure of watching it. Friends, let me also inform you that the world's fixed supply of water in all forms, that is vapor, liquid and solid, is enormous. About 97% of the Earth's water is found in the oceans and is too salty for drinking, growing crops and most industrial uses except cooling, and the remaining 3% is fresh water. Now friends, let me make you familiar with the fact that there are two different sources of water, which are as surface water. The water on the surface is called surface water, like lakes, rivers and ice. And the water under the surface is called groundwater, like rain, snow and dew, which sinks into ground as underground reserves. Friends, you must also learn that surface water can become groundwater through infiltration, while groundwater can become surface water through exfiltration. Surface water and groundwater, both of these are part of the hydrological cycle, which can be defined as the continuous recirculatory movement of the water of the earth. While being part of the hydrological cycle, the surface water and groundwater appear to end here, and they can differ in two important ways. Surface water is completely renewable, usually within days or weeks, while groundwater is not completely renewable, since it may take decades, centuries, or even longer time to renew. And fresh surface water is scarce, particularly when compared with the large volumes of fresh groundwater, which are known to exist below the surface. Now let us talk about the problems which are related with water resources. Excessive irrigation practices in semi-arid and arid regions can cause salt accumulation in soil and reduce crop productivity. And continuous depletion of groundwater along the coastal regions often leads to the movement of saline seawater into fresh waters and spoiling their water quality. About 40% of the world's population living in arid or semi-arid regions spend substantial amount of time, energy and effort in obtaining water for domestic and agricultural use. And the requirement of population depends on surface water bodies, thus results in decreased water table level, and which in turn also affects groundwater table. Now, dear friends, let me tell you the ways in which man is responsible for use and overutilization of surface and groundwater. They are as drinking, cooking, washing, domestic use, aquaculture and fisheries, industrial use, power generation, ecological conservation, and recreation. Now, let us talk about the uses and overutilization of water resources. With the growth of human population, there is an increasing need for larger amounts of water to fulfill a variety of basic needs. The overutilization of water occurs at various levels, like most people use more water than they really need. Most of us waste water during a bath by using shower or while washing clothes. And many agriculturists use more water than necessary to grow crops. Friends, agriculture also pollutes surface water and underground water stores by the excessive use of chemical fertilizers and non-toxic pesticides. Also, the industry tends to maximize short-term economic gains by not bothering about its liquid waste and releasing it into streams, rivers, and the sea. Now, let us talk about water utilization at global level. At global level, 70% of the water is used for agriculture, about 25% for industry, and only 5% for domestic use. Industrialized countries use a greater percentage for industry. 31 countries are already short of water, and by 2025, there will be 48 countries facing serious water shortages. The total annual freshwater withdrawals today are estimated at 3,800 cubic kilometers, twice as much as just 50 years ago. Studies indicate that a person needs a minimum of 20 to 40 liters of water per day for drinking and sanitation. However, more than 1 billion people worldwide have no access of clean water, and too many more water supplies are unreliable. Now, water utilization in India. India uses 90% of its water for agriculture, 7% for industry, and only 3% for domestic use. Around 20 major cities in India face chronic or interrupted water shortages. India is expected to face critical levels of water stress by 2025. Also, friends, there are 100 countries that share the waters of 13 large rivers and lakes. The UN has estimated that by the year 2050, 4 billion people will be seriously affected by water shortages. The demand for water has increased over the years, and this has led to water scarcity in many parts of the world. The situation is aggravated by the problem of water pollution or contamination. India is heading towards a freshwater crisis, mainly due to improper management of water resources and environmental degradation, which has led to a lack of access to safe water supply to millions of people. This freshwater crisis is already evident in many parts of India, varying in scale and intensity depending mainly on the time of the year. Also, with development pressures taxing the surface waters in many regions of the world, the trend has been to use groundwater to help or resolve perceived surface water scarcities. Without regulation, this trend places communities on a sure path towards groundwater mining. Now, a brief review about the exploitation and utilization of freshwater resources. First, let's talk about agricultural use. It's estimated that 69% of worldwide water use is for irrigation, with 15 to 35% of irrigation withdrawals being unsustainable. It takes around 3000 liters of water converted from liquid to vapor to produce enough food to satisfy one person's daily dietary need. And the second is industrial use. It's estimated that 22% of worldwide water is used in industry. Major industrial users include hydroelectric dams, thermoelectric power plants, which use water for cooling, and oil refineries, which use water in chemical processes, and manufacturing plants, which use water as a solvent. And also, water withdrawal can be very high for certain industries, but consumption is generally much lower than that of agriculture. Next is household use. It is estimated that 8% of worldwide water use is for household purposes. These include drinking water, bathing, cooking, sanitation, and gardening. Next is recreational use. It is usually a very small but growing percentage of total water use. It is mostly tied to reservoirs. Recreational usage is usually non-consumptive. Golf courses are often regarded as using excessive amounts of water, especially in dry regions. Next is environmental use. It's also very small but growing percentage of total water use. Environmental water usage includes watering of natural or artificial wetlands, artificial lakes intended to create wildlife habitat. It's non-consumptive but may reduce the availability of water for other uses at specific times and places. And next is increasing water scarcity. 50 years ago, the common perception was that water was an infinite resource. People were not as wealthy as today. They required a third of the volume of water we presently take from rivers. Today, the competition for water resources is much more intense. There is increasing competition for water from industry, urbanization, biofuel crops, and water-reliant food items. Now a brief overview about the exploitation and utilization of groundwater. Compared to other water resources, the groundwater has many advantages, like it has good quality because it is filtrated and cleaned in the unsaturated zone in the process of forming groundwater. And the investment of groundwater supply engineering is very little, but the efficiency is fast. Friends, groundwater is the major source of drinking water in both rural and urban India. Besides, it is an important source of water for the agricultural and the industrial sector. Water utilization projections for the year 2000 put the groundwater usage at about 50%. Till recently, it had been considered a dependable source of uncontaminated water. Groundwater crisis is not the result of natural factors. It has been caused by human actions. During the past 2 decades, the water level in several parts of the country has been falling rapidly due to an increase in extraction. The number of wells drilled for irrigation of both food and cash crops have rapidly and indiscriminately increased. Friends, India's rapidly rising population and changing lifestyles has also increased the domestic need for water. The water requirement for the industry also shows an overall increase. Intense competition among users, agriculture, industry and domestic sectors is driving the groundwater table lower. Now, let us talk about effects of overutilization of water resources. Now, the effects are as number first is lowering of water table. Excessive use of water for drinking, irrigation, industrial and commercial purpose has resulted in rapid depletion of water resources in various regions, leading to lowering of water table and drying of wells. And the second one is regional groundwater depression. Friends, because of the overexploitation of the groundwater continually and with high intensity, the groundwater resource cannot be recharged in time and the depression cones are enlarged, which even leads to the draining of the aquifer, reduction of water yield of a single well or discard the wells as useless. And the next is subsidence of land surface. The overexploitation of the groundwater not only causes the water level to decline greatly, but also the sediments in aquifer become compacted. It is called groundwater subsidence, and huge economic losses may occur due to this phenomenon. Next is seawater invasion. It mainly occurs in the coastal regions. The reason is that after the larger exploitation of the groundwater, induced gradients may cause the migration of salt water from the sea towards the freshwater sources, hence making it unusable. And the next is ground collapse. The overexploitation of the groundwater in bedrock region will lead to some geological phenomena such as ground collapse and fissure. And pollution of groundwater. Overexploitation of the groundwater not only quickened the infiltration of surface water but also the pollutants. So the groundwater gets polluted. Once it happened, it's difficult to recover. And also soil desertification. The continuous decline of groundwater level will lead to the degeneration of vegetation and destroyed environment, which is one of the reasons of soil desertification. And waterlogging. When excessive irrigation is done with brackish water, it raises the water table, gradually leading to waterlogging and salinity problems. Now friends, let us talk about the role of an individual in conservation of water resources. As we know that every individual has a major role in the conservation and equitable use of natural resources for sustainable lifestyle. Without natural resources, the survival of human beings is impossible. Different natural resources like forest, water, soil, food, mineral and energy resources play a vital role in the development of nation. Now, let us talk about the measures to conserve water. Water resources can be conserved by turning off the tap while brushing the teeth, washing the clothes, cleaning the vessels, and taking one bucket of water for bathing purpose and instead of using 2 or more buckets. Filling the water only to the level required for your clothes in washing machines. Watering the plants in your kitchen garden and lawns in the evening when evaporation losses are minimum. Using of drip irrigation systems in agricultural fields. Now friends, let us talk about the important sustainable water management methods like building several small reservoirs instead of few large dams and developing small catchment areas, and it protects the surrounding wetlands. And also soil management. Afforestation permits recharging of underground water, preventing leakage from dams and canals, preventing loss in municipal pipes, and excessive rainwater harvesting in urban environment, and water conservation measures in agriculture, such as using drip irrigation, and also desalination of seawater and groundwater to make it fit for human consumption. Increased irrigation efficiency in agricultural lands by reducing water wastage, and reduction in domestic water wastage by constructing wastewater treatment plants and recycling the treated water. From the discussion, it can be concluded that water is the most abundant substance on the Earth's surface. Proper assessment of the availability of water resources from surface and subsurface sources is crucial for its proper planning, development, and efficient management. Overuse of the water resources in our modern society is resulting in fast depletion of these resources and several related problems. Therefore, friends, conserving water often requires social change in addition to technical change, and the average person can usually do much to conserve water. That's all. Thank you.